Good evening, ladies and gentlemen. If you are joining us for the first time, you are very welcome. This is the Apologetics Academy. Our website is apologetics-academy.org. We meet every Saturday online at this time. Normally, it's 8 p.m. Greenwich Meridian time here in the UK, or 3 p.m. Eastern, 2 p.m. Central, 12 noon Pacific. And each week we bring on different scholars, experts, scientists from across the theological and philosophical spectrum to present on topics of interest to the field of Christian apologetics, particularly topics that pertain to the issue of whether Christianity is true. We also cross-examine and critique other worldviews such as Islam and Mormonism. There's a number of ways that you can participate in the discussion this evening. You can simply sit back, watch, enjoy the show anonymously. You can also participate in the chat window. You can also submit questions anonymously in textual form by hitting the Q&A button and typing in a question that will be submitted to our speaker for discussion later. And you can also click the raise hand button, in which case I will promote you to be a co-panelist with our guest speaker, and you'll have the opportunity to dialogue and discuss the matters at hand. Please be respectful of the speaker, even if you disagree vehemently with what he has to say, and also please keep your questions on topic. Just to give you some idea of what's coming up in future weeks, next week on Saturday, February 11th, we have Dr. Timothy McGrew, who's a well-known philosopher at Western Michigan University, and he is going to be talking to us about the reliability of the Acts of the Apostles. And then the following week, on February 18th, we have an atheist by the name of Ed Atkinson coming on to talk to us about why I am not persuaded by the resurrection of Jesus. And then the following week, we actually are, the Apologetics Academy will not be on that week because I am teaching it a weekend away here in the UK. So bring us back to this evening then. We have a friend of mine who's also an atheist, biologist by the name of Dr. Zachary Moore on to talk to us about the molecular evidence for evolution. We've had a number of pro-intelligent design scientists, biologists on in the past, but we've never had scientists who will offer us a critique of neo-Darwinian, or give us a positive case rather for modern evolutionary theory. So we're very thankful to have Dr. Moore on to discuss this with us. Zachary, do you want to maybe just for those that haven't come across you before, you want to just give us maybe a brief introduction to who you are and then just launch into your presentation? That would be awesome.
Sure. Thanks, Jonathan. And thanks so much for inviting me to participate. I know this is a makeup. I do a lot of traveling for work, and so my schedule is sometimes unpredictable. And fortunately, it worked out that I was able to do it this weekend, and Jonathan generously made time for me. So thank you very much. I think that the whole Apologetics Academy enterprise is really fascinating. I'm really glad that Jonathan is doing it. I rarely get to participate in the discussion, but I usually do follow along when the video is posted online. So hello to anybody who's following along after the video's been posted. Thanks for watching anyway. So yeah, so my name is Zachary Moore, and there's, aside from just my name that my parents gave me, there's a couple other descriptors which are maybe of interest here to this discussion. At the tail end of my name is a PhD, and the PhD is in molecular biology. I earned that degree at the University of Cincinnati in 2005. Strictly speaking, the program was pathobiology and molecular medicine, but that's a bit of a lot for people to recall, so I usually just say molecular biology and leave it at that. So I'm not exactly an evolutionary biologist. I'm not trained in the same way that Ken Miller was trained or Richard Dawkins was trained. But I do know a little bit about it, and it was necessary to learn for my degree, and just to be a biologist in general. You have to understand that concept. So that's sort of where I'm coming from scientifically. On the other side of my name is the word atheist. And I do want to mention that and chat a little bit about that just in general before we get started with the science. It's interesting to me that the fact that I'm an atheist needs to be mentioned. And of course, I'm not criticizing Jonathan. Of course, here in an apologetic situation, there are Christians, there are atheists, and it's helpful to know who is on sort of which side of the fence. But that being said, the concept or the discussion about evolution to me doesn't really take on that atheist theist coloration. So I'll just give a little bit of a background about myself. I was raised in a Christian home. I accepted Christianity. I considered myself a Christian. I was saved. I felt I had a relationship with Jesus Christ that was very meaningful to me. And all this while I was studying biology, while I was studying in graduate school. I held my faith sort of close to me, and I never felt particularly threatened learning about evolution. In fact, there's a story that I can recall. I think I was in about 7th grade, perhaps, and I was taking a class at church where we were learning about Satan and Satan as a deceiver. And somehow in my head, and I don't know exactly how this happened, but somehow in my head, it occurred to me while discussing Satan that, maybe all this stuff that I've been reading about, all these the stories about fossils and these other discoveries, paleontology, etc., maybe that was some satanic deception. And I don't know why that bubbled up in my brain at the time. I never really worried about it before, and my parents had never really given me any cause to worry about it before. Certainly, there was no anxiety that they displayed or that they instilled in me with regard to sort of creationism versus evolution. They encouraged me to learn as much as I wanted about any subject, really. But I can recall approaching my father sometime later and just testing that idea with him and asking him, what do you think? Is there some possibility that Satan has screwed up, the fossil evidence? And my dad didn't really respond to me. I can recall he just sort of looked at me sideways and rolled his eyes. And that was all I needed to know to sort of say, what, this is probably not a good avenue for me to explore. This is probably not fruitful. So I didn't really, think anything of it after that. And so the entire time I was a young Christian, going to university, learning about biology, learning about evolution, I never considered it to be an attack on my faith. And it really it never would have occurred to me to wonder about what an atheist might have to say versus what a Christian might have to say. But of course, now in the midst of this discussion and in this context, of course, you just can't not note that I do happen to be an atheist, but maybe a little bit more than that. And the reason I think for this sort of focus on the religious context of this question, it goes back a long, long time, easily 100 years or more. Of course, there's, here pictured is Clarence Darrow and William Jennings Bryan at the Dayton, Tennessee, quote unquote, Scopes monkey trial, where the question was put forward, are we concerned about the Rock of Ages or the Age of Rocks? And I think even 100 years after that, that's still kind of the question that is focused on that. But it's important to realize that even that trial was itself a manifestation of the tumult that was going on in Christianity at the time. This really wasn't a scientific trial or discussion or conflict even. Right. So as Darwin's ideas began to be published and be widespread, there is little serious conflict in the scientific community. But what happened was in the culture, people started to understand that these ideas, and not just Darwin's ideas about evolution, but it could be argued that this trial was much more a function of the development of higher criticism of the Bible more than anything else, which started in the 19th century and really prompted a complete reinterpretation of the entire seminary landscape in the United States. Princeton Theological Seminary completely flipped and became liberal and sort of modern, and that's what caused the sort of fundamentalist backlash, and then which eventually brought in William Jones Bryant to that, and thus this conflict over evolution can be seen as a function of the fundamentalist backlash to the modernist movement within the church, right? So this really isn't even necessarily an atheist onslaught on the church. This is an internal discussion within Christianity. But of course, in this day and age, the way that it's presented is very much an atheist versus a Christian situation, right? So instead of Clarence Darrow and William Jones Bryant, now the two sides of the issue are Ken Ham and Richard Dawkins, and Ken Ham is a fundamentalist Christian, a young Earth creationist. He has, I guess it's a reasonably successful enterprise in northern Kentucky, very close to where I grew up. And Richard Dawkins, likewise, has had a very successful career as an ethologist and then a popularizer of evolution, and now I don't know exactly what you want to call him, but some sort of an advocate for atheism in some way. And so these are the two sort of most visible figures in the culture war, and one is a fundamentalist Christian of the most fundamentalist persuasion, and the other is let's say a fundamentalist atheist of the most fundamentalist persuasion, and they're arguing over evolution. So that's why we've sort of got this all tangled up with who's an atheist and who's not. But I do want to point out that there are I should have had a picture of Ken Miller there. It disappeared. There are many Christians for whom this is not an issue, and I'm including my former Christian self in that. But I'm thinking predominantly of Ken Miller, Dennis Alexander in the UK, and Francis Collins, and these are evangelical Christians, right? They are very clear about their identity as Christians, and they are also very clear about their acceptance of evolution. Now, to what degree God plays a role in that is sort of up to further debate. I think they all certainly do think that there is a role for God in that, so we don't have to chuck him out in the bin entirely. But they also don't see a conflict between their Christianity and evolution, which was also the case for me. And in fact, the title there, Do We Have to Choose, is from Dennis Alexander's book of the same title, which I have an autographed version of. I got to meet him in Fort Worth several years ago, actually. Wonderful, wonderful gentleman. So let's move on to evolution then. All right. So what's the big deal about evolution? Why is Richard Dawkins and Ken Ham, why are they so at odds about it? Why are they so passionate about what the answer is? Well, it's an old story, and I'm not going to tell the entire story of evolution, but it starts with this, really, or a part of the story starts with this. So this is a fossil of an animal which is known as Glyptodon, and this animal lived in South America millions of years ago, as much as 20 million years ago. And fossils of this animal were discovered by Charles Darwin when he was visiting the continent. It's possible that they may have been discovered earlier than him, but he definitely became aware of them and described them. And he also noticed this animal, the armadillo, and we have lots of these here in Texas where I live. I normally see these sort of crushed in little bits on the side of the road where they get run over at night because they don't have very good eyesight. Every so often you'll see a live one sort of scuttling around in the bush. But Darwin asked himself this question. Well, we've got this one type of animal, the glyptodon, that clearly doesn't exist anymore. The only thing we have are fossil remains. There were no more living glyptodons. But we also have the armadillo, which seems to be kind of the same thing. And just looking at the two of them begs the question, are they related? And if so, how? How would they be related? And the question began to be asked, well, maybe there's some way of describing how one species became another species. Maybe they have some common ancestry. Maybe the one is ancestral to the other. And he applied the same idea to the finches that he explored on the Galapagos Islands, which are quite a bit more famous than the glyptodon. And that's all very interesting as well. But the real question, right? So as human beings, we are predominantly concerned with things that are of interest to us, right? So it's all well and good to sort of chat about animals and how animals may have come about. But the real question is, what about us? What about humans? Are we animals? I mean, are we subject to the same biological forces as other animals? And if so, if these other animals have evolved, if the glyptodon and the armadillo are related, if they're related through some ancestral line, what does that say about humans? If we're animals like them, have we evolved? And if we have evolved, who are our closest relatives? If we look out sort of at the animal kingdom, what sort of animals might we suggest would be closely related to humans? And this is kind of where it gets a bit uncomfortable. And I'll tell another quick story. So I've got a good friend of mine who's a evangelical Christian, and he was actually for a time he was the youth pastor at his church. And he invited me over to his house one time. They just finished like some sort of I can't remember if it was a Bible study or some event with the youth group there, but they were all sort of hanging out at his house. And I came over and I was chatting with a few of them, and he mentioned to me this girl, and he said, "Oh, I know, Zach, that you're really interested in evolution. Let's just say her name's Linda." He said, "Linda has been studying evolution in school, and she's been telling me all about it and how fascinated she is by it. And I thought you might like to know that." And I said, "Yeah, that is pretty cool." And I sat down with Linda and I was chatting with her for a little while. And she was sort of matter of fact about the whole thing. Like, "Oh yeah, we studied it, and I don't know. I mean, I've heard that it's controversial, but nothing really seemed that controversial to me. Everything sort of hangs together. It makes sense. The evidence seems to line up." And I was a little, taken aback. I am here in Texas after all. I'm not used to the idea that kids here would be learning straightforward about evolution. And so I asked her, I said, "So wow, that's really cool. And you don't really have a problem then about the idea that humans are also animals and have evolved and share some common ancestry with these other animals?" And she said, "Oh no, I don't accept that at all." And I said, "Well, that's really interesting. That's curious. If you don't mind me asking, why? So you don't have a problem with anything else? You don't have a problem with understanding the evolution of all these other animals. It's just humans that you have a problem with?" And she said, "Oh yeah, of course. I can't accept that." And I said, "Well, why?" And she said, "Well, because humans were specially created. They were made in the image of God." And I said, "Oh, okay. So you don't have any scientific, sort of criticism about human evolution specifically?" And she said, "No." I said, "So it's just because in your mind this contradicts some basic theological belief that you have?" And she said, "Yeah." I said, "Look, that's fine. As long as you know that it's a function of your own theology, that you don't accept this about humans, then there's really nothing more that I can do. I'm not prepared to argue theology with her. If she had something scientific to share or to discuss, then that would be a different situation. But I think her position is similar, I would say, to a lot of the objections about evolution. I think if we took humans out of the equation, I don't think there would be quite as much anxiety about it. But because of the implied contradiction between our evolutionary origins and, let's say, a fundamentalist interpretation of the first few chapters in Genesis, I think that's what is really causing the bulk of the cultural conflict that we have about it. So whenever I discuss sort of common descent, I like to sort of bring this up as just an example. So what you see here is my family. At the top, this is my father. On the right, on the left is my mother, and down at the bottom are myself on the right and my two younger brothers. Now, we are all related, or so I think we are. At least that's the story that I was given. I haven't actually tested that, but we can suppose that we are related. And what does it mean for us to be related? Clearly, I'm related to my brothers in kind of a different way than I'm related to my father and my mother. And in the sense that my brothers, I don't have an ancestral relationship to my brothers. I have a common ancestry with my brothers, right? So we both, or all three of us, have descended from the same pair of human beings. But let's say that I was feeling particularly, I don't know, anxious or skeptical about what my parents told me. And let's say that I believed for some reason that my father was not my biological father. Let's say I believe that I was actually descended from somebody else entirely. Well, how would I determine that? How would I verify that? Well, in this day and age, it's fairly easy. We can do a paternity test. And what do paternity tests look for? Well, they look for genetic sequences that are held in common between two individuals. And if there are a sufficient number of those held in common, then a common or a descent pattern can be inferred, because we know that this is how species reproduce. As individuals have children, they pass along their genetic information, right? So the genes that I have are a roughly half and half admixture between my mother's genes and my father's genes. So the information that's inside of me came from someplace. It came from both of them together. And if myself and my brothers are found to have the same basic admixture of the two individuals at the top, then that's a molecular confirmation of our common ancestry. Well, let's take it forward a little bit. Let's say that it's myself and 4 other people completely at random. Let's say, well, maybe not completely at random. I did, I stuck Jonathan up there just for the hell of it. Some of my ancestry, I had mentioned this to Jonathan earlier, some of my ancestry does go through Scotland. So it's possible, in fact, it's possible that we could be distantly related. But let's say I took 4 other people completely at random. Well, how would I determine how closely I am related to them? Well, I would probably do the same sort of testing pattern. I would want to look at our genes. I would want to look at the different sequences that I have, different markers that I might have in my genome, and see how closely they line up with everyone else in the group. And we can then infer that if I have more sequences in common with any of these people, then I'm much more likely to be more closely related to them. And we're more likely to have a more recent common ancestor than anyone else in the group, right? So out of all the people that we see there, it's likely that unless I'm related more closely to Barack Obama's mother's side of the family, it's more likely that I'm more closely related to Jonathan. And the molecular evidence would bear that out. And none of this is particularly controversial, right? We do paternal testing as a matter of course. It's accepted in courts of law. we're doing it more and more often now that sequencing, technology has become incredibly easy and relatively cheap. Everybody should be doing this to some degree or other. So let's push it out a step further. Now I've chosen 4 other species entirely. Well, we know these species have DNA. We know they have sequences that are similar to ours. How would I determine which ones are more closely related to me? I do the exact same thing, right? The same thing as if you took 4 random humans and pulled their DNA and did an analysis. All you would do would be compare the sequences, compare different markers, how many we have in common, and then we could conclude that that the ones that are more in common have a more recent common ancestor. Again, this only becomes controversial at this point. And it only becomes controversial at this point because now we're talking about other species. And now there are some Christians for whom this becomes a point of theological anxiety, which in my opinion, it doesn't necessarily have to be. But this is the progression, right? So we go from, okay, of course, yeah. Ooh, I don't know. So what I want to go through with the following slides is just a survey of how we start to evaluate this and how we can build a case and verify that the ancestry relationships that we have, that we are quite frankly are implied by the fossil evidence, are implied by the physiological comparison between different species, et cetera. All these things that had been, and still are really, the traffic of evolutionary biology. We can get to the same point just by looking at the molecules. But first, we have to understand what the molecules are. So I'm going to go through really quickly a basic molecular biology 101 in just a couple minutes, just to make sure that we're all on the same page and we know what these terms are. So when I talk about the molecules in molecular biology, I'm really talking about 3 basic types of molecules. There's DNA, RNA, and protein. All right. Everybody's heard about DNA, I'm sure, and probably everybody's heard about RNA. And protein is the stuff that bodybuilders eat, to get big and strong. But really, these are the fundamental molecules that do pretty much everything for us. So DNA is the template molecule. It contains all of the information, the instructions for everything that's going to happen in our bodies, essentially. It's the template molecule. And RNA makes a copy of that template and takes it out of the nucleus and basically turns it into the functional molecules. And RNA can do some other things as well, but that's essentially what RNA does. And then the functional molecules are then the proteins. Those are the molecules that do the things that our bodies do, that make it possible for us to have all the different functions that we do. So DNA, RNA, and protein. And these are not just words or acronyms. These are molecules, and molecules they're very, very small, but they have structure. Right? And I want to get this across that there's a particular structure to DNA. Most people are familiar with sort of the double helix shape, and the fact that it's a kind of a sequence of nucleotides. RNA also is a sequence of nucleotides, but it's very typically in different shapes. It's still kind of helical. It's still kind of reminiscent of DNA, but it can conform to many other different shapes that have different functions, and I won't get into all of those. And proteins have a wide variety of shapes. A protein can be pretty much anything in 3 dimensions, and it's important to get that across for the points I'll be making later. Is that all these molecules can be represented as basically strings of letters, but in actuality, they are things. They are physical things that exist in 3 dimensions, and their existence in 3 dimensions has relevance to their function, especially proteins. Nucleotides, just to bring everybody up to speed, is these are the components of DNA, represented here by the bases. So it's basically a ribose backbone with a different base that they get paired up, A and T and G and C. Adenine being the A, thymine being the T, guanine G, and cytosine C. If you've seen the movie Gattaca, they have a lot of fun with A, T, C, and G. So those are the nucleotides that we're talking about. So again, when we're showing these letters as sort of strings of information, we're really talking about these physical molecules laying out end to end. Amino acids, these are the components of proteins. So proteins are, again, strings of letters as we represent them, but it's a polymer of amino acids laid end to end with different functional units. And we've got here, there's just 4 represented: isoleucine, asparagine, tryptophan, and tyrosine. And you can see that they're all different shapes, but they have sort of commonalities between them. Now it's important for you to understand the concept of a codon. So I've been talking about representing nucleotides as a string of letters. And the way that the string of nucleotides is converted or translated into a string of amino acids is that 3 nucleotides in a unit are read by the machinery that translates it. And so you have a long chain of letters there right in that row, but that chain gets broken up in 3s. So instead of just CCA, TTT, CAC, ACT, etc., you've got CCA, TTT, TCA, CAC, etc. And so each one of these 3 units, these 3 nucleotide units, becomes a codon, is read as a codon, is read as a unit. And so the CCA is read by the machinery and eventually becomes is translated as a proline amino acid, so that the string of letters that we see at the top, as represented as nucleotides, becomes a similar string at the bottom, again represented as 3 letters, PRO, PHE, SER, for the individual amino acids, but we can also represent them just by single letters, PFS, HCP, etc. So a string of letters that represents nucleotides can becomes eventually, through translation, transcription, translation, becomes eventually a string of letters that represents an amino acid chain. And that amino acid chain it can be functionally a protein. And that's by parsing the nucleotides as codons. Now, there are 4 amino acids, or I'm sorry, there are 4 nucleotides, A, C, T, and G. And since the codons take the form of 3 nucleotides in a row, that means that there is a code of sorts. And each codon corresponds to an amino acid. And because there are 4 amino acids and 3 nucleotides possible. I'm sorry, I'm screwing that. Because there are 4 nucleotides and 3 nucleotides possible per base, that results in a code that has 64 options. And you can see on this diagram, if you start with the first base in the codon on the left-hand side, and then step forward with the second base along the top, and then within each group of 4, you add the third base, either TC or G, you get 64 different options of codons, and each codon then corresponds to an amino acid, as you see. And that's the genetic code. And this is the genetic code for almost all life as we know it. There are some other single-celled organisms that use slightly different codes, but this is pretty much it for, certainly for the primate family. We all use the same code. Now, you'll notice, of course, there's 64 codons, but there's not that many amino acids. There's only really 20 amino acids that correspond, which means that there's quite a lot of redundancy. And the redundancy means that you can get the same amino acid with lots of different codons, sometimes as many as 5 different codons in the case of leucine. And for most of these, it's at least two. I think the only one that has just the one is tryptophan, or no, methionine. Methionine and tryptophan both. Well, methionine is the start codon anyway. But so there's redundancy. So you have options. You don't have to use AAT to get asparagine. You can use AAC, right? And you don't have to use 3 T's to get phenylalanine. You can also use TTC. So that's redundancy, and there's redundancy that exists among the codons. Now, there's also a level of redundancy that exists among the proteins. So the function of the protein is itself a function of its shape and chemical properties. And there are many amino acids that have similar physical and chemical properties to other amino acids. So you can swap out, for example, in many cases, isoleucine for leucine. So if you see those represented there, all it's really done is move the position of one of the methyl groups. But it hasn't changed the charge at all, hasn't really changed the shape or size. So it's essentially a neutral shift if you were to shift an amino acid chain from having a leucine to an isoleucine. And so because there's functionality that can be achieved between, and if you change an amino acid, you change the sequence, so it becomes a different protein in sequence, not necessarily in function, which means that there's protein functional redundancy as well. So you don't necessarily have to have the same exact sequence to get the same function from the protein. So that's redundancy that exists at the protein level because of similar amino acids. Now, there's also redundancy at the level of the nucleotides. And as shown before, of course, the DNA itself, there's 4 different nucleotides, which means that there's 64, because they're read in groups of 3, there's 64 different codons that are possible. And there's only 20 different amino acids synthesized, which means, as we saw when we looked at the genetic code, that there's lots of redundancy at the level of the genetic code itself, which means that you don't necessarily need the same nucleotide sequence to get the same protein, to get the same amino acid sequence. And for any average amino acid sequence of just 10 amino acids, you could write that 1,000 different ways in nucleotides. And the longer the amino acid sequence becomes, the more options you get in terms of nucleotide sequences that are possible that would have the exact same outcome. It would create the exact same protein in that organism. And so this is functional redundancy at the level of the sequence. So 2 levels of redundancy. There's the protein functional redundancy and the sequence functional redundancy of the nucleotide. So here's the argument. And I have to mention here Douglas Theobald, who put this together at Talk Origins. He I think he did this over 10 years ago. And I reached out to him one time. I asked him if he would be interested in like developing it more, doing like a podcast series based on it. And he said, I don't have the time, so you just do with it whatever you like. And so I did use it for my Evolution 101 podcast, which some people listening or watching might be aware of. But anyway, so this is the formal argument. So we know that there are many genes which are ubiquitous. Ubiquitous means that they exist in pretty much every type of life that we know. They're kind of essential for life. You really can't have life without them. And the function of the gene products, it's really independent of the particular species, right? It doesn't really matter whether the species lives in the ocean or lives in a desert or lives on a mountaintop. It doesn't matter whether it's a mammal or a reptile or whatever. They've got to have this type of gene to create this specific product, which is essential for life. We also know, and this is what I've been demonstrating over the past few slides, is that the sequences are functionally redundant at the level of the nucleotide and also at the level of the protein. So we don't necessarily need the same protein sequence in every organism. We certainly don't need the same nucleotide sequence in every organism. Therefore, having any sort of similarity between different species within their sequences, whether at the level of nucleotides or at the level of the proteins, is unnecessary. We don't need that. And one of the main reasons why we might expect to see similar sequences is on the basis of heredity. And we've already established that we can sort of appeal to similar sequences, similar genetic markers between different members of the same species, certainly, in order to determine common ancestry relationships. And so the argument goes, if we can do that within species, we can do that without species as well. And if we see the same correlation of similar sequences, then we can infer then a close common ancestry. And so if we want to look at those ubiquitous genes, we can then infer a genealogical relationship, or accept the hypothesis of a genealogical relationship, a common ancestry. So let's look at some of this. So one of these ubiquitous genes is cytochrome C. And cytochrome C is, it basically functions within your mitochondria, and it is essential for oxidative phosphorylation, which is basically how we live. It's how we make energy and keep ourselves alive. And so cytochrome C is essential for this. Virtually every species, from Homo sapiens, which you see at the top in the chart there, Homo sapiens is at the top, that's us. And at the very bottom, you see Saccharomyces cerevisiae, also known as yeast, which we can thank for the whiskey we drink, and beer, and wine, and bread, and lots of fun things. And in between, there's lots of different species, and they're kind of an order of similarity to the human sequence. So underneath Homo sapiens is Pan troglodytes, or the greater chimpanzee, Bos taurus, the cow, Canis familiaris, dog, Mus musculus, mouse, Rattus norvegicus, which is the rat, Danio rario, which is the zebrafish, P.Z. Meyer's favorite organism. And at the bottom, as already mentioned, the humble yeast. Now, if we look at the sequence, so what's displayed here is a section of the cytochrome C gene in all of these organisms, and they're lined up. I used blast analysis to line them up, and just showing you a section of it here. But if you line up the amino acid sequence of this protein in all these different organisms, what you see is the human and the chimpanzee sequence is exactly the same. Exactly the same. And as you go sort of further away from what sort of fossil analysis and other analysis would predict would be a closer relationship to human beings, you see more disparity in the amino acid sequence. So what's the likelihood that this would happen just sort of randomly? Well, it turns out that can be measured, and it's a chance of 1 out of 10 to the 93rd power that this would happen randomly. So some evidence in support of that being a common ancestral relationship. Interestingly enough, so you have at the top, you have with the human version of the protein, you have a very different protein sequence than what you see with the yeast. At the bottom, you've got the yeast, and the yeast is only similar to the human version by 2 amino acids, at least in this section of it. So it's fair to say the human version and the yeast version are very, very different, but they still have the same function. And this is demonstrated here by this experiment published in 1999. And this is a common thing that's done in molecular biology labs called complementation, where they take genes out of model organisms, usually yeast, and they replace them with the human version and see if it works. Well, in this case, it did. They took out the cytochrome C from the yeast, and they replaced it with actually a chimera version of the gene that incorporates human DNA. And it got a functional response. So you put human DNA in yeast, and you replace the cytochrome C that's there, and it's clearly a different gene, but it has the same function, so that you've got functional redundancy. So you don't necessarily have to have the sequence that humans have. You don't necessarily have to have the sequence that yeast has. The most important thing is that you have the function. And there's obviously, if the human version works just as well or almost as well as the yeast version, at least in the yeast. I don't think we've performed the opposite experiment, taking yeast and put it into humans. But that demonstrates functional redundancy. So there's no pressing reason, or there's no biological reason why you would need to have the one particular sequence in one organism versus the other. And yet we do see this relationship where the more similar sequences are in the organisms that we would predict to be more closely related to us. So now let's look at the same gene, but we're not looking at the protein product, we're looking at the nucleotide sequence itself. Well, if we look at that, we're looking at the same model species. We see pretty much the same breakdown, right? So as you go from humans at the top to yeast at the bottom, you see more and more differences incorporated in this section of the genes. And in fact, it's not at all surprising because there's 10 to the 49th different potential nucleotide sequences for this small, relatively small protein. And yet, as I mentioned earlier in the previous slide, there's no difference at all in the human and chimpanzee proteins. The amino acid chains are exactly the same. There is a difference in the nucleotides. So the chimpanzee nucleotides has changed by 4. But those are essentially neutral mutations, or they could be mutations on the human end as well. But the difference between the two is 4 nucleotides. And but those are neutral, and it does not affect the amino acid sequence. So now we've got 2 levels of redundancy that both show this relationship, that predict an ancestral relationship where the chimpanzee and the human are more closely related than the cow, than the dog, et cetera, et cetera, et cetera. So if the humans and chimpanzees are not descended from a common ancestor, then that would be it would just not be predicted from this data. It would be very a rare thing for that to happen randomly. Okay, so those 2 bits of evidence are looking at the sort of the coding region of the genome. Now we're going to look at the quote-unquote non-coding region, although the non-coding region is not necessarily a non-active region. This just doesn't code for actual genes. And it's actually a significant portion of the human genome, as it is for most mammalian genomes, actually. I think, I've got 45% of our genome is non-coding DNA made up of DNA transposons, 13% LINE, 20% SINE, and 8% long-term repeats, which also includes some endogenous retroviruses as well. It's what they used to call junk DNA. It's not really junk DNA, it's just non-coding. They're sequences which move, they move around a genome. They can either copy and paste, or they can cut and paste. And it depends on the mechanism, whether it's the one or the other. But the important thing is that these are changes that occur within the transposon. They may not necessarily be selected for. They may just be happening in the background. But wherever the transposons move to, that still gets incorporated within the genome, and the genomes are inherited, which means the novel transposon locations are inherited. So that's something else that we can look for. And in fact, these are, quite frankly, when we look at paternity tests, we're mostly looking at these types of elements. So this is a reconstruction, a phylogenetic reconstruction of primates. They were looking at transposon insertions. They focused on the short interspersed nuclear element ALU, which is very, very common. And they wanted to look at specific insertions of this transposon in different primate species. And on the left-hand side, you can see a couple DNA blots. Basically, this is different PCRs that they ran. You see the bands that are tracking at the higher weight. So there's on the top, it's 500 base pairs versus 185. So you see the higher one in the human lane, but not in the other primates. So this is an insertion. So basically, when the insertion happens, it makes the DNA sequence longer and therefore weigh more, and it tracks slower through the gel. So in the human lane, you see that it's got the higher band, the heavier band, which means that the insertion has happened there. So for that particular insertion, that is specific to humans. It does not exist with bonobos, chimpanzees, gorillas, orangutans, etc. But another one, a completely different one that you see on the bottom, and I've cut out some of the other ones that they published because they went through basically all the different options. You see that the insertion exists in humans, bonobos, chimpanzees, gorillas, everything except for the, I think, the New World monkeys. So this is a specific insertion at a specific place in the genome. That's the only way that it could work via PCR. And so what this means is that there are transposons that exist in these different genomes that are held in common. And if you list out all of these insertions and you sort of line them up by which ones are in common between which species, on the right-hand side, you can reconstruct a phylogeny. And the phylogeny constructed by, and this is just on the basis of the molecular evidence, not on the basis of anything else, just on the basis of that, it shows, again, what we would predict based on other evidence, that the Panpaniscus, which is the bonobo chimpanzee, and the Pantroglodytes, which is the common chimpanzee, are more closely related to each other, but they exist in a clade with Homo sapiens, which is us, and we are then, as a group, related to gorillas, and then what's the other one? The big orange one. I just lost the name. Pongo. At any rate, so this reconstructs that phylogeny, which is exactly what we would predict based on other lines of evidence, but we don't necessarily need that because this is just going on the transposons. So in addition to transposons, other sort of non-functional or non-coding DNA that exists in our genome is pseudogenes. And pseudogenes are previously functional genes. They used to have some function. They used to work and do something. But they've since become broken for various reasons. They've either had been recombined with some other portion of a chromosome, perhaps, or they've undergone some mutation or deletion or some insertion that screws up how the exons and introns line up, or there's all sorts of different ways. But this happens, and we know that it happens. In fact, the reason why humans have to get vitamin C in their diets is because we have a gene that makes it, but that gene has been broken and is now a pseudogene. So we have to get vitamin C from our diet. In cats have sweetness receptors that are more or less identical to ours, but they've been broken, and they now exist as pseudogenes. So your cat really can't taste sweetness. And again, a pseudogene, a broken gene, is it's not necessarily going to be eliminated from the genome unless, for whatever reason, it's strongly selected against. So as long as it's not selected against, the progeny are still going to inherit it, right? So humans, even though our vitamin C gene was broken, it didn't matter because we were getting it from our diet anyway. So there was not a strong selection against not having a functional vitamin C gene because we were eating plenty of, or our ancestors were eating plenty of foods that had it anyway. So it didn't really matter. But the evidence is still there. And if there's another gene that has undergone a pseudogene transformation in humans as well as in chimpanzees, so the 21-hydroxylase gene, we actually have a couple copies of it. So one copy of it has undergone that pseudogene transformation. The other one still exists, but if that gene gets mutated, then it can cause disease and other bad things. But if you look at the sequence and you line up between the human sequence and the chimpanzee sequence, which you see here in this figure, the humans is HOSA, and then PATR is chimpanzee, GOGO is gorilla, and POPY is orangutan. Gosh, that's the other one, orangutan. But if you look and you see this version of the gene, the asterisks all the way to the right-hand side, you see 8 asterisks in a row. That's a deletion. So there was a deletion of 8 nucleotides in the human pseudogene, in the human 21-hydroxylase pseudogene, and there's the same exact deletion in a corresponding 21-hydroxylase pseudogene in chimpanzees. And this strongly suggests that this was inherited from a common ancestor, from whatever the common ancestor was between humans and chimpanzees before our populations separated from each other. That pseudogene transformation happened in that gene, and we both populations have inherited that. And finally, the evidence from endogenous retroviruses. And these are kind of cool, interesting. Retroviruses in general are interesting. Viruses in general are interesting, I think. Retroviruses are viruses that use reverse transcriptase in order to actually insert themselves into the host genome. So they don't just come into your cell and make a mess. They actually come in and insert themselves into the host genome. And depending on the type of the tissue that it gets inserted into, that it might be something that you have to deal with, for a little while, or maybe the rest of your life. But it's not necessarily going to be something that you pass along to the next generation unless that insertion takes place in a germ cell. So if it takes place in a cell that gives rise to a spermatocyte or an oocyte, then if that happens, then that insertion will be passed on to the next generation. So you start to see the incorporation of these endogenous retroviruses, and some of them remain active as viruses, but very often, because there's not a strong selective pressure to keep them functioning, they become effectively pseudogenes as well, or pseudo little microgenomes within your genome. So they stop working, they stop making virus, and your genome can even incorporate it to do other things and to function as the regulatory element, for example. Whichever is the case, it doesn't really matter. The point is, it stays in your genome and the sequence can be tracked. And there was a study done, going on 15 years now. Again, this is similar to the analysis done looking at transposons, but this is looking at endogenous retrovirus insertions. And they basically lined up all the endogenous retrovirus insertions in different primate genomes. And they tried to calculate in which species did they find the most similarities between all these genomes, and using that analysis, they were able to construct a phylogeny, which you see here, which is, again, pretty much exactly the same as what we saw with the transposon analysis. We see humans and chimps sharing a recent common ancestor, and then us as a group sharing a common ancestor with the gorilla, and then the orangutan, and the gibbons, and old world monkeys, and new world monkeys. So this is, again, evidence in support of the common ancestry between humans and chimpanzees. So in summary, the evidence from molecular biology, we have protein functional redundancy and DNA sequence redundancy. These are very similar concepts, but they work at kind of two different levels. But they both reinforce the idea that it's absolutely not necessary that any two given species have the same sequence in order to get to the same end. Right? So there's a lot of flexibility. So there's no reason at all for us to expect a common sequence, or any sort of, there shouldn't necessarily be any sort of relationship that implies common ancestry based on the common sequences because of that. And yet we do see that nonetheless, which supports the hypothesis of common descent. And then in the non-coding region of the genomes, we see transposons. When we reconstruct those, it shows a phylogeny of common descent. We see pseudogenes, which also shows common descent, and finally endogenous retroviruses, which also show common descent. So everything that we can look at in molecular biology supports that hypothesis. So I'll end it there, and I guess we open up for questions.
Thank you, Zach. Do you want to just unshare your screen so we can?
Sure.
There you go.
There we go.
Folks, this is the interactive portion of the program. If you would like to interact with our guest speaker, Dr. Zachary Moore, then simply click the raise hand button at the top of your screen, and I'll promote you to be a co-panelist with him and give you the opportunity to interact. You can also submit questions using the Q&A button if you would prefer to enter it in textual form. We have two hands raised at the moment. We have Krishnam.
Krishnam.
Hey, Krishnam. Are you there? Okay, I don't know where Krishnam is. I'll bring up the other hand just now.
Oh,
James, you're on.
James.
yeah, great. Hey, I was wondering about the statistics you mentioned before about how unlikely it is to XYZ exponential unlikelihood. How much do we really know about how genetic switches affect those statistics? Is there a chance that those might change in the future as we discover more mechanisms?
Well, yeah. So those assume that so that the exponential likelihood of that happening is assuming that that's going to happen by random chance. So that's sort of the upper bounds of the likelihood of that happening without any other factor influencing it.
But if we found that one switch can kind of cascade to other switches, would those numbers decrease or change in any way?
Yeah, I mean, if it could be demonstrated that it was anything less than, yeah, absolutely. So what I'm saying is that is that's the upper bound of likelihood, given the lack of any other influence, right? So obviously, if all these animals or if all these different species have common ancestry, it's not. There's not this exponential likelihood that we would see those. We would expect that, right? So the probability of that goes down tremendously.
Okay, thanks.
Yeah.
Okay, thank you, James. Krishnan, are you with us at this point? Okay, evidently not. So I'll put him back in the audience. We also had a couple submitted questions. Let me read those. Krishnam says, who created DNA, RNA, or proteins to start with?
I don't know. Yeah, it's a good question. So I'm not really an expert on that. I won't really get into that. But suffice to say, the boundaries of this argument specifically assume only that we have the evidence that we have, right? So we know that DNA exists, we know that RNA exists, we know that proteins exist. Given that we know those things exist, what then can we infer from that available information? So I don't know that we can infer who made what, but given the information that we have, common ancestry is, I think, what we can infer.
Okay, we had a hand raised from Sharif. Sharif, with us.
Hi, Zachary.
Hi.
Nice to meet you. I really enjoyed your talk. That was like really great stuff. And I came in a little bit late, so I'm sorry if I asked things that you already have discussed. Is my mic on? Can you guys hear me?
I hear you just fine, Sharif.
Okay, great. So I came in at about the point where you were making the argument for where the objections are coming from in the community of faith regarding common ancestry. And it seemed like what you were proposing as the rub was about the theological basis of the objections to common ancestry based on a particular interpretation of Genesis 1, etc.
Right.
And I wonder if you were open to the idea that there might be evidence-based objections or methodology-based in terms of how we infer.
Oh, sure. Yeah. It's just that's not by. So the story that I told was not that. The story, the young woman that I was chatting with, she had been. I'm pretty sure it was high school level. But she'd been learning about evolution, and everything she said, everything made sense to her. All the evidence seemed to point to that as like the obvious conclusion. The only thing that she pulled back on was whether or not humans and other animals had common ancestry, because she believed theologically that humans are created in the image of God and specially created. And so for her it was a purely theological objection. I'm not saying that that's the case for all Christians. For myself, as when I was a Christian, I didn't have any objection to it at all. So I was able to understand the theology of Genesis within an evolutionary framework, and it didn't bother me at all. So
okay, well, I didn't hear the beginning of that anecdote about the person, so that's really good to clarify. So I had a few points. I really like how you constrained your model of how you came to the way to infer from the sequence information that common ancestry is the best conclusion to common similarities of sequence. And if I can make sure I understood it properly, what you were saying is that since there's all these levels of redundancy in the coding of function, in the encoding of function in a cellular system, let's say, going from protein to whatever that protein the role of that protein in the system, you don't have to have the exact amino acid sequence.
Right.
So there's wiggle room there. And then going back to that, you have codon degeneracy, so there's wiggle room, and even where DNA sequence you can get to encode even the same protein sequence. Okay, and so that seems to, I like the clarity of those constraints, but what it sets up and what I'm wondering what you do with this is if you're making some assumptions about the appearance of degeneracy or the appearance of, what do you call it? Redundancy. All right. So what I mean by that, if hopefully it's clear, but what I mean by that is that as long as we don't understand why the sequence is exactly what it is in the DNA to encode that particular protein, then we can take a posture of, well, it could be anything because we know what the code is and we could change it. But that, I would argue, is like a function of the gaps argument, because we just don't know why the sequence is that. And if at some point we understand that there is actually another layer of control on the system as a whole that is embedded in what particular codons you pick to encode that particular amino acid sequence, then what that would tend to do to your argument is to weaken that as a basis for inferring common ancestry from common sequence. And I can give you an example, which is that we now know that across all kinds of species, there's a thing called codon bias, which is that not all, so the genetic code, there are many genetic codes, but we can leave that to the side for a second.
Right.
But the codons that are used to encode particular amino acids, the distributions is different across different species. And there was a recent paper that was really cool where people looked into what functional aspect that might have. And they found that there, in some cases, was a direct link between a particular rare codon being used, for example, which, to just sum up with that, the implications of that is that there would be a pause in the protein synthesis machinery while you're waiting literally for a molecule of tRNA to get in there that's at low concentration in the cell, for example. And because it's this, okay, and then what does that do? That they have found has been correlated in particular cases to a difficult part of the protein to fold that would need more time. And therein you see that there's this kind of, where it looked like there was the appearance of redundancy, it's actually something functional where the limitations on the protein folding process or the idiosyncrasies are accounted for in giving that sequence more opportunity to fold properly. So in those types of situations, what I am concerned about in that methodology that you set up or that inference logic is that it has to take steps back as we understand more and more about why the apparent redundancy.
Mm-hmm.
What do you think about that?
No, that's a very good point. Yeah. And there might be exceptions to the general rule, right? So there might be reasons why you would want a particular codon in certain situations versus others, let's say. But we also know that, and this was also maybe at the beginning of my talk, the analogy that I used was how do we determine like paternity, for example, right? So I put up an image of myself with my family, and what if I was skeptical that I was actually descended from my parents? You know, they tell me that I am, but how do I know that? Maybe. So how would I check? Right? How do I confirm that I actually have that common ancestry? Well, that all depends. It all is based on the assumption that similar sequences or more similar sequences implies a closer genetic relationship, right? And so if we apply that to like paternity tests, we can also apply that to human genome analysis, like how closely related are different populations on the world. Then we can also apply that to different species, right? And so it's not just about that there's codon redundancy. We're also seeing a pattern of similar sequences.
Okay, now that's a really good point. So with that, I don't accept that premise that we can apply that paternity test model to different sequences. So I think that what strengthens our ability to infer and to infer paternity. So like say if you doubted your parents are actually your parents, okay, and instead you have this other potential cause for your existence, which is that you were implanted here by aliens and they just happened to give you a genome that looks similar to these two people, right? So there are other ways to explain that, but we also have the evidence that every time two humans have mated, they have made another human, and that is also, I think, building the case for this is a probably better, more what's the word? I don't know, feasible inference to make. Whereas what I would challenge is that if when they're doing genetic testing across species, they're not doing the same exact thing that you're doing within species. And so here's an example, like so I don't know exactly how to argue this, but one thing that's confusing to me is that I feel like you already start begging the question a little bit when you introduce the concept of species without right? So why put it in there? To go from your set of pictures of humans to your set of pictures of you plus those other like non-human primates, what's the purpose of calling them different species? Isn't that sort of arbitrary? Why aren't they just variants of the same population?
No, that's an excellent point. So let's say that we did that. Let's say I wasn't presenting them as different species. Let's say I was presenting them as so the first picture of me and my family, I just say, this is me and 4 other organisms. All right. And then the next picture is, this is me and another 4 organisms. And then the third picture is me and another 4 organisms. Yeah. Now, how do I determine, when we know that when 2 organisms mate and have progeny, that they inherit sequences from the parents, right? So how do I determine how closely I'm related to any given organism? Any given organism. I don't care what species, and believe me, the concept of species is problematic for lots of other reasons.
I know.
All we're just saying is these organisms. How do we determine that? How do we determine how closely related I am to any of these given organisms?
Okay, well, I think that there needs to be more detail added to the constraints on our inferences from looking at sequence. So if we can all agree that I arrived here through descent with modification from my parents, and so did every other human. But now we have a disagreement. We're at an impasse about whether that's also true of chimpanzees and whether we have common parents. And so the question is, what kind of modifications are extant, observable within this human population, within the chimpanzee population? And those have to be the limitations on the types of modifications that you can infer happened to lead us to this this proposed split between our proposed common ancestor. No?
What are the what limitations? What do you mean?
So what I mean is like, okay, I'm trying to apply the uniformitarian method of historical science, which is to say that you have to constrict yourself to causes in the present, known causes in the present, to infer as the effects of causes in the past,
okay.
The causes of effects from the past. And rather than, because if you don't do that, right, you could propose any kind of force or event and nobody would be, you could just say, well, anything can happen. And then that's not science, right? That's not a viable scientific method, mm-hmm. right? And so, so what I'm saying is that if within the human population, we see like single nucleotide polymorphisms, we see insertions, we see deletions, we see gene duplications, we see like all these different things that are part of the standing variation among within this population. Uh-huh. And then we see a type of modification that cannot be explained by the way that those types of modifications within our population arrived. Then we have a new mechanism that we don't see happening here. We would have to infer a cause that we don't have any evidence is currently going on within this species to create diversity in this species. And you can, and the only way to get a new species is to have some kind of a variation mechanism that allows that branching off to happen. So it would be very like specious or whatever to say, well, there was that mechanism in the past, the branch off happened, and then mechanism went away. That's not the uniformitarian method. So is that clear?
Well, I don't think anybody's proposing that, and so I don't know. And again, I'm not making a case for speciation. I'm making a case for common ancestry.
You cannot have speciation. You can't have one without the other. You're gonna have to explain to me how that works.
Well, we have common ancestry among humans, right?
Right.
So again, all I'm saying is from organism to organism.
You're proposing universal common ancestry, though. You're saying that we have common ancestry with non-human primates, and then you call them different species. So you must be saying that speciation is a part of how that.
But I'm not concluding that they're a separate species necessarily. Like I'm just using a representative of that population. So species is just it's just a concept, right? It's just a label that we put on things.
Okay, well that's I see. So when it's allowed to be just a concept, I mean, I feel that that's not very rigorous. You know, like if you're not willing to like nail down a definition that can then be tested as to whether it is coherent with reality or not, then that is not very useful scientifically.
Yeah, well, that's the problem of species. That's the problem of species. It's a problem. I've got something on YouTube about that problem of species.
I'll look that up. I'm really interested in this problem. Okay.
So forget about species for now. Just talk. We're just talking organism to organism, right? So if I'm talking me to you versus Jonathan, how do we determine which one of you I'm more closely related to?
Okay, what we would do is look at, what has been done. This is one method, I suppose. Unless we had absolute knowledge of the family tree of humans, then we could look at the SNP profiles and assume that those SNPs represent actual historical events that are changed, that led to changes in the DNA from a previous state or sequence. And so from those sets of inferences build a model wherein if your SNP profile looks closer to mine, then that would mean that you and I were closer to the originator version of us, which yes is yet another question there.
Right. So we're comparing sequence similarity. When you talk about SNPs, you're talking about single nucleotide polymorphisms.
I am, but you know there's a term called substitutions, right?
Mm-hmm.
What do you make of the use of that term? Why do we have it? Why do we need it? Or do we need it?
I think the critical concept here is sequence similarity, right? So if you have a sibling, your sequence is more likely to be similar to your sibling sequence, yes? Than any other person that exists.
Sure.
Right. So that's not controversial. All I'm saying is that when you're comparing on the basis of sequence similarity, no matter how that sequence is similar or different.
Yeah, and so go ahead. Sorry.
So you can infer a closer ancestry, a more recent common ancestor, right? On the basis of similar sequence.
But I'm trying to. So there's another potential reason for a similar sequence than just similar ancestry or common ancestry, and that's common design. How do you?
Okay. Yes. So you already mentioned this. What if an alien designed me in a laboratory and created my genomic sequence for whatever reason to be exact or to look like exactly like what my parents is? So if they let's say, I don't know, if they got a sample of my dad's DNA and my mom's DNA, and they worked in a lab and there was some sort of genetic engineer of some sort, and they created me in a dish with half of my mother's DNA and half my father's DNA, and then surreptitiously implanted me in my mother, and she gave birth to me thinking that I was hers, but in reality I'd been specially designed to look like I was hers.
Okay, but in that particular case, you do see how there's a problem that you just took material from both parents and just did the same thing, but in a dish, and that's
however you do it. Whether you do it by taking material from the parents or whether you do it de novo, doesn't really matter. However you're engineering the DNA, right? So if an alien was engineering my DNA in a dish to make it look like I was descended from my father and my mother, but I'm not actually, how would I know?
How would you know? I think this is a great thought experiment.
Well, the answer is you wouldn't know.
Right.
The answer is there's no way to distinguish between whether that sort of a thing happened versus whether I actually was born from my parents.
Right. So we would constrain our inferences in that case, like I was trying to say before, based on our uniform and repeated experience of how life on Earth appears. And
right.
Enough eyewitness experience of babies being born from procreative activities that the most parsimonious explanation would be that, not the aliens, but scientifically we would not know whether it was aliens.
Exactly. And so I would exactly. So there's no evidence that you could appeal to in that point to prove or disprove that possibility. And so the only thing you can appeal to would be Occam's razor.
Well, okay, do you feel like Occam's razor is totally objective? Like the most simple explanation to an atheist? No, seriously. Like I believe that if you are a priori committed to a materialist cause for something, then for you, Occam's razor is going to be committed to a materialist cause.
I'm not a priori. So I was a Christian, and I had no problem with this. I was not a priori committed to materialism.
Right now, are you committed to materialist causes for life and biological information?
Not a priori, no.
What do you mean when you say that?
So where I am now, I've come to from a very long process. So yes, I do find materialism persuasive now, but it's not something that I stick in the ground and stand on.
So you're open to a conscious mind as being an agent in the production or instantiation of genetic material de novo from raw materials of dirt and energy.
I'm open to it philosophically, but my point is that there's no way that you could confirm or disconfirm that hypothesis scientifically, right?
Well, then why are you committed to common ancestry?
Well, because it makes the most, because it, again, I'll appeal to Occam's razor, because it's the simplest explanation of the evidence.
But it does, okay, to you it may be. So what I'm saying is that as more evidence comes in, though, you have to admit that the grounding on which your methodology is standing is sort of like becoming more and more shaky, because as long as it is true that we can infer common ancestry from this, because this degeneracy is real, then you're A-okay. But I feel like that is the same argument that people make when they're like, oh, ID is a God of the gaps argument because you just haven't given science a chance to find out how it was done without God. And that's not true. It's a positive argument. But in this case, you have your whole thing is based on not having a rational intentionality behind why the degeneracy is what it is, that there's no design in it. And as we find out through science and through investigation that it's not true, then eventually I feel like that has to show you that that is not, that Occam's razor is not pointing towards that conclusion.
So I'll say this, if there is evidence that leads to that conclusion, then I will happily accept it.
Well, that's good. Well, we can have more discussion. I don't want to let somebody else come in here and thanks for engaging.
All right, thanks. And Professor McClatchy, please give Sharice an A for the day.
I love A's.
We have currently one hand raised. If Sharice, you want to continue after that question, then feel free to raise your hand again and I'll be back up.
Okay, I'll keep eating my soup.
Okay, we have a question raised, hand raised from Tim Hull.
Tim Hull.
Hello, Zachary. Can you hear me?
I can hear you, Tim.
Fantastic. My camera's not working for some reason. My green light is on, but I'm not showing up, so I apologize.
I'll just look at Jonathan.
He just look. Well, Jonathan just put his picture up, so I don't know. Anyway, I'm not nearly as scientifically sophisticated as the last person was. I apologize.
No worries.
So my question would be, you had mentioned that there was this probability, 1 to the 10 to the 93 thing at one point. Is that correct? I know my chat just left.
There was a couple different sort of calculations of probability that a certain situation would happen by random chance. Yeah. Sure.
So you're discounting any sort of, guided force, ID, anything like that, preloaded or not, or quantum indeterminacy, God using quantum indeterminacy to kind of direct things along the way with those numbers, correct?
Yeah, I mean, I would love to if I could measure that, then yeah, I could incorporate that.
Sure. Okay. So that just added a question there. Okay. So then the other question I had is at the split, at one point you said, if we're looking at our ancestry, at some point we were, related to, you're using your calculations to say that we're related to chimpanzees or, some other primates, right?
Yeah, that's correct.
So if we take the current mutation rate and go back to the point where that divergence happened, is there enough time for us to be able to get the amount of changes that you laid out in your graph in order to be able to have the diversity that we see right now in humans?
Well, so the difference between chimpanzees and humans is negligible. It's 1% or less than 1%, depending on where you look. So there's really not a lot of differences genetically between humans and chimpanzees.
Hold on, when you say 1%, we're talking about billions of characters, though, right? So 1% in a billion. I mean, we're still talking hundreds of thousands, if not millions, of genes that have to be changed.
Oh, sure.
Over a certain period of time.
Well, so every, roughly speaking, every generation is born with about 700 mutations. That's just the background mutation rate.
Sure.
So mutations are always happening. It's the best guess, I think, to when the split happened, I think was, I want to say 100,000, 200,000 years ago, maybe earlier. I can't remember exactly when it was. Let's say 1 million years ago. It's really not, there's nothing about the mutation rate. And the molecular clock is a complicated issue in and of itself. It kind of depends on how you measure it. It depends on what you're looking at. The different chromosomes have different mutation rates. For example, the Y chromosome, as well as the mitochondria, has different mutation rates. So it depends on what you're looking at. But generally speaking, no, not to my knowledge, does that give anybody pause about whether or not there's enough time. The real question is, I think, or the more interesting question is, given the relatively short amount of time, I mean, geologically speaking anyway, and the fact that there's so little variation between humans and chimpanzees, why are we so different? You know, why do we not have hair on our bodies, for example? Why do we walk upright? Why are our brains so much bigger, et cetera? So that's the real question, and the answer is I don't know. There are when the Human Genome Project was first published, which was administered, I should point out, by Dr. Francis Collins, who's an evangelical Christian. When that was first published, that was one of the first things that they wanted to look at as soon as they had the chimpanzee genome sequence as well. That they wanted to see, so what's actually different between the two of us. And so far, I believe we still don't know. There's a couple regulatory elements that seem to be very highly modified in humans as compared to the chimpanzee genome that are expressed in the brain, right? And so one of the big differences between humans and chimpanzees as species is the way that our brains work and the way that we use them. And so it sort of fell into kind of what people were looking for. They were looking for some massive change in the brain to say, well, this is why humans are so much smarter than chimps. But as far as I know, we still don't know.
Okay. So you're basically, I mean, I think I caught, again, that there are these changes that you were talking about. We don't have hair all over us, you know, more of our skin is exposed, our brains are bigger. And they're really just, you know, given the accepted amount of mutation rate per generation, we just don't know why it has changed how it has in as short a time frame as we've seen.
No, we don't. We don't know. We don't know specifically what. So this is really the question. What could you do genetically to a chimpanzee to turn it into a human? And the answer might be nothing, right? So of course, you know, when we're talking about common ancestors, we're not necessarily talking about chimpanzees, right? We're not saying that humans came from chimpanzees. We're saying that humans and chimpanzees had a common ancestor. Now, what was that ancestor like? It was probably more like a chimpanzee than like a human. It was. It did not walk upright. It had a small head, had a small brain. It had probably had hair on its body. So it was definitely more chimp-like than human-like, but it was not a chimp, right? So chimpanzees and humans are cousins. They're not our parents. So yeah, I don't know, right? So asking that question, it's an interesting thing to consider. Like, you know, what could you do? Could you tweak something in a chimpanzee to make it more human? Maybe. I don't know. It's kind of like what they every so often they talk about getting one of these mammoths that's frozen up in Siberia and harvesting some of the cells and putting one of the nuclei into an elephant embryo and seeing if you can give birth to a mammoth. You know, would that be a mammoth? Not exactly, because it's in an elephant embryo. It'd probably be something close, but I don't know exactly what it would be. So a lot of these, I mean, these are good questions, interesting questions. I don't know that they're answerable yet.
So then doesn't that sort of undermine your presupposition or the conclusion of your argument to say that then there is this, you know, common ancestry? If we're saying, well, we don't know what the ancestor is, but we're just kind of guessing that there might be an ancestor, you know, on some level, it seems like we're begging the question. We're saying we can't get humans from any kind of ancestor, but we're assuming on some level that there are cousins, and we don't know what changes needed to happen in order to get from there to here. But right. We're pretty sure that changes did happen for us to get from here to there, and we don't know how long it took us to get from here to there, but it did. So there's like lots of maybes and assumptions in there to reach that conclusion that you were like, and there's common ancestry with nothing else going on. background.
So it's not an assumption that's any different than the assumption that we would make if you and I compared our DNA and determined that we had a common ancestor someplace. Like, I don't need to know who it was necessarily. I don't need to know that it was my great 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 grandfather to know that we had a common ancestor.
Other than the fact that we know that there didn't need to be any specific special type changes between you and your great 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 grandfather in order to get you, as you're suggesting, to say, well, we know that when we look at a chimp and we look at a human, they're somewhere close, so we're inferring that same thing. I feel like if you can't answer the question, how do we get from a chimp to a human, you can't do that, because there's no question on whether or not you came from a human, even 20,000 years ago. But when you start just making that bubble bigger, 150, 200, 6 million, 10 million years ago, you've then gone beyond what you can reasonably say is the ancestor, correct?
So what you're appealing to, the same thing that Charisse was appealing to, this species boundary, and the concept of species is just a concept, right? So it's a concept that we give to put different things in the buckets. It goes all the way back to Linnaeus, and really, for honest, all the way back to Aristotle. It's a human concept. It's a way that we think about the world. It is not necessarily something that is demonstrable by science, right? So whether or not there's a species boundary between us, don't really just think of it in terms of organisms, right? So you and I are two organisms. We can compare our DNA and compare our DNA to some other third group, some third organism. And we can, between those three, we can determine which one is more closely related to the other, right? And so this is all we're doing. Instead of you and me and Jonathan comparing our DNA, it's me and a chimpanzee and a gorilla comparing our DNA. It's just three different organisms. It's the same thing.
Right. And like I said, I'm not nearly as scientifically adept as you guys are. But my understanding was that there was there's some lines as far as who can mate and produce offspring that then create these lines. So like, I am very different than a cat or a dog or even a gorilla in the sense that we may have similar genetic makeup, but again, that seems like then we're just begging the question and saying, well, because we're carbon-based life forms in a similar environment, i.e. this floating planet that's just hanging out 93 million miles from the sun, we have to have these common building blocks. We see these common building blocks, therefore we're going to assume that they must have a common ancestry.
Right. Okay. So yes, you can't mate with a cat, even though you might try really hard.
Nope, won't do that.
And that is, generally speaking, that is a widely accepted parameter for what separates one species from another. But again, if you look for my talk, The Problem of Species, it's imprecise, and it's not as clear of a line as we would necessarily like to believe.
Okay, no, that's fine.
And in fact, we can hybridize. In fact, didn't they just hybridize a human cell line with a pig cell line? They're going to be making. I think they're going to be trying to raise human organs inside pigs now. So they've got a human-pig hybrid, which fans of Archer is somebody's excited really over there at that show.
Sure, and it was all designed in a laboratory, right?
Yeah. But the point is that the boundary is not absolute. That's the point that I'm making. It's not absolute.
Right. The only other question that I have is, what are you drinking?
So this is Johnny Walker Black Label.
Oh, okay. Nice. I bought some. I bought something which I thought was good for Jonathan, but Jonathan is a far better human being than I am, and he does not drink scotch.
Oh no, a Scotchman that doesn't drink scotch.
I know. Sorry, Jonathan. I'm calling you out, but I'm supporting you, bro.
We'll have haggis together sometime.
I used to have a whiskey group where we would all pitch in and buy really expensive whiskeys and try them, and it was.
I've got Lagavulin over there someplace in the back of my cabinet.
Oh, nice.
Glenrothes Glen Ross has a few really nice vintage, or I don't know what you call it exactly.
Have you ever had a Glenfiddich?
Mm-hmm.
The 12 and the 14? Those are pretty good.
12, but not the 14.
Yeah, the 14's obviously pretty good. It's a little aged longer. But there's also another one. There's like a Japanese whiskey that's actually really good.
Yamazaki?
Yamazaki, yeah. There's
two
Yamazakis that are really
not quite as good as this, but man, it's decent. It's solid.
Yeah. Okay, I'll get off. Thanks.
All right.
Thank you, Tim. We have two hands raised. The first of whom is Chris Klaus. Chris, go ahead.
Klaus.
Thank you very much, Jonathan, and good to talk to you, Dr. Zachary, as well. I just had basically one comment, and I was just going to get your opinion on this comment. That's all.
Okay.
And it says here that biologist Boy Britton and other evolutionists continue to assess the result in terms of evolutionary theory, speaking about the chimpanzee DNA and how it's decreased. Years ago, it was said to be about 99 to 98%, and it's actually lowered to less than even 95% of the genetic material, of course. So it continues to the theory, but in fact, there is no scientific reason to do so. The theory of evolution is supported neither by fossil record nor by genetic or biochemical data. And on the contrary, the evidence shows that different life forms on Earth appeared quite abruptly without any evolutionary ancestry, and that their complex systems prove the existence of an intelligent design. Now, would you have an opinion to that, sir?
What are these? What organisms are supposed to have appeared spontaneously? Do you know?
Not quite sure off the top of my head. It's so are you saying that there has been never? Are you saying that there's never been life forms on Earth that appeared quite abruptly without any evolutionary ancestry?
Well, it kind of depends on what you mean by abruptly. So they may be talking about the Cambrian explosion. And the Cambrian explosion is a period. It's a geological period. I think it's like, gosh, again, I'm not a paleontologist, but I think it's like tens of millions, maybe hundreds of millions of years long. But in the grand scale of time, it's like a blip, you know. And they called the explosion because there's all these different forms of creatures that appeared in the fossil record relatively quickly. And again, relatively quickly is relative. We're talking at least tens of millions of years of time. And there were no sort of predecessor fossils to them that were in the fossil record. One reason for this may be that at that point in time was when organisms developed actual body parts that could be fossilized. So prior to that time, there were very few organisms with actual hard body parts that could be fossilized. If they're talking about the Cambrian explosion, that might be what they're referring to. And that, I guess, would be my response to that.
Alrighty. Now, I was also looking up as well, and it's stated that nearly 75% of our human genes have some sort of counterpart to nematodes, which is a millimeter-long soil-dwelling worm.
Yeah.
Now, this definitely does not mean that there is only a 25% difference between man and these worms. According to the family tree made by evolutionists, the Chordata phylum, in which man is included, and the other, which is the worm, were different to each other over 530 million years ago.
So you're asking, are we related to worms?
And by the same measurement that we look into chimpanzees or type thing, isn't that a logical question when 75% of the genes are associated to worms as well?
Yes, we are related to worms. We're related to, as far as we know, maybe the archaebacteria are a little weird and we're not quite sure where they fit in. But yeah, for every animal that you've ever seen in your life, we're related to it. And plant.
Okay. Thank you very much for your opinion on the matter.
Yep, and in regards to archaebacteria, Zachary, it's thought that they are our most closely living prokaryotic relative, as I'm sure you know.
Oh, really? So they're actually they're more
they're believed to be closer to us than bacteria. This is actually what my research is
regular prokaryotes. Okay.
Charisse.
Charisse is back.
She's back.
Hi. So I wanted to pick up on something that you said regarding the claim that it's the same exact method of inference that you use for within humans to infer common ancestry of outside of humans. One of the things that you haven't really discussed much is like genome structure. So if I'm looking at a SNP profile, I don't have any information about the relative locations of those genes in the genome in that SNP profile. So what I'm talking about is, like, for people who may not know, like SNP, single nucleotide polymorphism, you have mutations, insertions, deletions, and they're basically signatures of change from one generation to the next over time.
Right.
But what's done with that is the way they do that, to detect those, it doesn't depend on where they actually are. So if you had a scrambled genome, for example, where if you think of the gene sequences as cassettes that, let's just say Craig Venter in his power went and created an organism that had a human that had all the same genes, just in a different order. That process of detecting the SNPs would not detect that, right?
So wait, okay. So you're taking an entire genome and you're randomizing all the. Okay, so you're randomizing all the. So you take all the chromosomes and you chop them up into bits and you throw them up in the air and you put them back together randomly. And all it hasn't screwed up any of the genes.
Right.
All the genes
or any of the like regulatory regions. Somehow you've luckily just taken things out in a way that let's presume, in fact, I don't believe this is true, but let's presume there's no information encoded in the particular order of, gene cassettes and things like that.
Right.
The point being this, that who cares whether that thing was screwed up or not? My point is that the methods that we use to detect the SNP profiles would be blind to that event, to that randomization.
Yes, that's true.
Good, great. Now, moving on from there,
okay.
Wouldn't you say that if you encountered a population of organisms that had all the same genes and things as humans, but just in a totally screwed up order, wouldn't you need to find an explanation for how that reordering happened if you were to infer common ancestry between those populations?
Okay, so you're saying that there's a human-like organism out there that has all the genes that we have.
Sure.
It's just its chromosomes have been put through a blender. How do we figure that out? How do we figure out common ancestry between humans and the
Not
blender?
how do we figure it out? We are assuming there must be because of common sequence based on your parameters, right? They have all the same sequence. It's just in a different order. What we have to explain is how it got in that different order, do we not?
Yeah. I mean, I don't know that there was anything like that. Well, so actually there is one thing that's kind of similar to that. Believe it or not. So chromosome 2, the human chromosome 2. Are you familiar with that?
Yes.
Okay.
Go ahead.
Well, just for the benefit.
People want to hear about it.
So human chromosome 2 is actually, it actually looks like two chimpanzee chromosomes that have been cut and pasted together to make a new chromosome. So humans have 23 pairs of chromosomes and chimpanzees have 24. So where do they get the other pair? Well, it turns out that other pair most likely was combined to make our human chromosome 2.
Okay, so here you have an example where we looked for evidence to explain something that had to have a historical event attached to it.
Okay.
You see 23 chromosomes. It is not why? Okay. It is not it's not part of the standing variation in the genetic makeup of the human population to have a variance in chromosome number.
Oh yeah, it is. Are you kidding me?
Of viable people who are fertile and who are, tell me who these people are who have different number of chromosomes than 23.
XXY.
Huh?
People with XXY. There's lots of different variations.
Well, how about non-sex chromosomes?
Well, okay, if we're going to put parameters on it, I guess. But my point is that we, within the human population, of course, we know that there's lots of weird things that happen with chromosomes.
Okay, we do, but aren't those usually like detrimental to fitness?
Yeah, and that's the case with lots of mutations.
Okay, but in this particular case, it seems that every single time well, I don't know about every single time because I'm inferring there were other times that failed, and I have no evidence of those times, but in your narrative, those times exist. But whenever you have a change in the chromosome number, right, you seem to get a new, what we would call species.
Yeah. No, not necessarily. Again, you're stuck on this species concept.
I'm not stuck on it. Darwin is stuck on it.
Well, we've all been stuck on it. But so what would make it a different species? It would only make it a different species if whatever happened to the chromosome caused some barrier to gene flow, right? If it resulted in some new population that did not share genes within another population, then that would create conditions that would result in a situation that we typically refer to as speciation. Yes. Does that happen often? No. I mean, I can't think of any time that's happened recently within the human species, although we've only been looking at genes for the past 70 years. Okay,
so that's good segue to my other, I think it's an important question to ask. Do you think that speciation is still happening in the human population?
Yeah, I got that question all the time when I was doing my podcast. That's what people want to know, and it goes back to that point. We don't really care about all these other animals. We want to hear about what's happening to us.
It's not that we don't care. It's that it's where the rubber meets the road. And to me, whatever you say with humans is applicable to zebras. It's just that we don't get all hot under the collar when we're talking about zebras.
Exactly.
So let's talk about humans.
Exactly. As soon as it affects us, we're like, how dare you? So yeah, is speciation happening? Well, speciation happens when you have two different populations that can't share DNA. Right? Mm-hmm. Eventually, if they get to the point where the changes that accrue between them mean that they can't exchange DNA anymore, they are more or less effectively different species, even though they might look exactly the same. They might. So you've heard the concept of ring species.
Yes.
Okay. So ring species is like, for example, the Arctic gull. Like, you can track these different subspecies all around the Arctic Circle, and I can't remember the name of all the species, but they're up in Scotland. So Jonathan, you look for them. But if you track them, the different subspecies, but from one to the next, they can mate in between. And if you follow them around the Arctic Circle, the different subspecies can mate, back and forth until you get all the way back around to the Northern Canada species. They cannot mate with the Great Britain species. Now, what that means is you've got this entire population of all these different organisms that are different. They all look the same. They're gulls. They've got white feathers and gray feathers, right? You probably wouldn't be able to pick one out of a lineup, but when it gets back to full circle, that subspecies will not mate with the subspecies that started the circle in Great Britain. And so, okay, so right now they're all sort of connected, and there's gene flow that's possible all the way from one end of the globe to the other. But if something were to happen, like if, let's say, I don't know, if a meteor struck in Siberia and it completely destroyed one of these subspecies that was in the middle of the ring, then you would have two different species of gulls, and they're not exchanging genetic information anymore. And over time, we would expect them to become more and more different, especially if they were facing different environmental pressures. So again, it's very fuzzy. It doesn't really lend itself well to black or white, but that's when we would say, okay, that's when speciation is happening. Once one population is no longer able to exchange DNA with another population, that's when it's primed for speciation.
Okay, so then is speciation a ratcheted process?
Ratcheted meaning what? That there are certain things that like knock it forward?
No, meaning that you can go forward, but you cannot go back. And therefore...
So that sort of assumes teleology, right? So that assumes some sort of a goal, some sort of an end goal. And there's not really that, at least as far as we can determine, there's not really an end goal for speciation. Like, things are not trying to become something else. Populations respond to environmental pressures, and they also respond to just sort of the random happenstance that happens to them, just sort of weird things. But it's all in that moment. So if something gets pushed in one direction, it's unlikely that it's going to go back. I mean, you can have atavisms happen from time to time. But atavism is sort of the recapitulation of ancestral forms.
Okay.
You can have that, but that's not a general thing that happens. It's the population's change is determined on the environmental pressure. So if let's say for example that you had a dwarf species, let's say a dwarf elephant that lived on an island, and let's say that climate change was reversed and all the seas all the oceans got frozen up and the water levels went down and the island was now connected to the mainland. And this species of dwarf elephant went onto the mainland and became larger because it had a lot more room to go around, a lot more resources, and all the humans had died off. So there was nobody to hunt it or take its tusks. Well, then you might have that species change, like in size. It might still be an elephant. It might become something else. I don't know what. But let's say it stays an elephant, just gets bigger. Well, then let's say that some other weird thing happens and the climate becomes warmer and the sea levels rise again. And the species now gets trapped on that island again, and it becomes a dwarf elephant again. So I can see that sort of situation happening where a dwarf species becomes a larger size species and then becomes a dwarf species again, but only in response to the environmental pressures.
Okay, and in that case, we definitely agree. So you're saying that evolution is not ratcheted, or speciation is not ratcheted, in the sense that, in that example, did you create a genetic barrier or procreation barrier between the big ones and the small ones? Were there any left on the island, or there would have been some kind of...
Let's say that there's some left on the island, yeah.
Right, and then, maybe for reasons of practicality, there was a barrier of reproduction in that case, if there were dwarf elephants versus really giant ones. But in that case, again, there's a failure of the category. The boundaries of that category seem really irrelevant to the question at hand, because why would we call them different species just because, of physical problems they face with making babies? Like, that just doesn't really seem to suit the question at hand.
It's the tyranny of the discontinuous mind. That's why we force that category.
But how I propose this, that I agree that this category is obnoxiously, flimsy and whatnot, but I'm not willing to cede that this category, that a category of some kind exists, because I think that a better term maybe for it would be a form in the Platonic sense. I don't know. I'm not really like a very well-read philosopher at all, but
that's precisely the problem. Like the reason why we try to, it's Platonism. That's why we try to force them into these categories. It's the influence of Platonism.
But what if it's not wrong? What if it is necessary because it is true? Because that is reality.
But that's not what science shows us, and that's not the picture.
Science doesn't. You already said science can't.
That's not the picture of the world that science shows us. Science shows us ring species. That's not Platonic.
Science shows us ring subspecies.
But that's not Platonic. Yes,
it is, because the ring and all of its members and contingent groups are they all belong or are representatives of a prior form.
But then what happens when there's an explosion in the middle of that ring? And now
it doesn't matter.
Well, it does matter, because that's something that can happen.
It can, and it affects the history from that point forward, but it doesn't change the reality of the beginning.
But what's the form? So now you've got two forms. You had one form, now you've got two forms. That's not Platonic.
No. The form. So, I mean, this is my philosophical, challenge to this, is that the form has to be something that is immovable, otherwise it's useless. But, and yeah, it is the form was instantiated into matter, and I have to ask you about how you would define life, but so you create an organism, and by virtue of being an organism, it reproduces after that form, and from that point, you can have all kinds of variants. So all we're looking at now is variants and trying to interpret whether there are two causes for the variance, or one. We are trying to decide whether all the variants can be explained by descent with modification, or if some of it is explained by descent with modification and some of it is explained by common design and separate ancestry. That's the question.
I mean, so that would be interesting. I don't know of any way to test for that.
That's my next question. Do you think that there might be signatures of that are detectable? Signatures where that cause, if it is whatever the historical cause of a genetic divergence, that is the result, or genetic difference, that's the result not of descent with modification, but of a design event. Do you think that design event would be detectable? That would be detectable as the case?
No, that's a tough one. I don't know how it could be.
Okay, so are you familiar with like any intelligent design theory?
A bit.
So this is like the crux of it, right? The question is, are influences from conscious agents in the past detectable now in the present? So what we would have to do, using, like I said before, the uniformitarian method, is to understand something about what conscious agents do now, the kinds of effects they cause now, and the signatures in those effects, and then we can look for those signatures in things that exist now and infer that was the cause then. Yeah?
So what are the signatures?
That's good. Okay. One of the signatures is what's called specified information. Okay, what is that? So you talked about the genetic code earlier. Okay, and you said it was a code of sorts, and I was like actually in my living room going, no, it's not a code of sorts, it's a bona fide code. Okay, it really is a code. So essentially you've taken matter and it is now representative of sequence information in other forms of matter, and that is not it's not a priori necessary because of the physical properties of said matter. That sequence, that the DNA, or that the DNA sequence that encodes an RNA sequence that encodes an amino acid sequence, there's nothing about the properties of quarks and neutrons and all of that that you can build up from scratch from the physical properties of matter and say, oh yes, of course that's why ATG is methionine. Of course, yes, I see. There's none of that, right? It's arbitrary, which is the same thing we see when you encode language of any kind. Okay, so that's one property, is that you have sequence information that specifies some other property or function or type of information. And that is what minds do, is they invent language. And only minds do that. That whenever we see language, we always see that it came from a mind in our experience. So do you agree to that?
Well, I don't think it's a language, though. So you're making an inference that it's a language. I don't see that. So
what is the difference between a code and a language?
Well, a language has that's a good question. Language has like a message, like there's a communication, right? You're trying to it's a language is a sequence of abstract concepts that map to objects in the real world.
Ooh, that's good. Abstract concepts
But that's not what DNA is.
Hold on. Match to objects in the real world.
Mm-hmm.
Objects in the real world. Okay. So those objects in the real world. So I'm going to, that's not what DNA is. Okay. So I will, and are you stepping away from the idea of communication? You started to say something about communicating.
Well, so with language, you're trying to provide information about objects.
Ah, okay.
So look, if I say, I'm raising my glass, let's talk about me. So the word I is an abstract concept, but it refers to me. Am raising is an abstract concept that refers to this action. My glass refers to this object. Again, these are all just abstract concepts that have. I'm not a language expert, but this is.
Don't
just,
need to be.
So it's about concepts and objects.
Okay, this is so good. This is so good. So would you say that somehow in language there is a marrying of concept and object, such that the sound? Okay, and when we say object, can we also include some form of energy as well? Object could be matter and energy. So for example, I'm speaking right now, and so this is spoken language as opposed to written language, as opposed to sign language, right? But all of those things are happening in the physical world. I'm changing the energy in this room, which is being picked up by this computer and transmitted. It's all transducing one form of energy to another and getting to your ears, right? I could also send you a letter, and that's an object.
Why you're so focused on energy?
Because I want to include that, because matter and energy, I want to put them together as one thing, as separate from meaning.
Matter and energy as separate from meaning.
Yeah, meaning that when you marry the concept and the message or the meaning, that's what I would call instantiation. That's like you are saying, this means that, and you are doing that, and you're creating that happens in the context of a language system, and there's other components, right? So I agree, it's an analogy. It's not exactly the same thing that's happening in biology per se, because we are two minds that are manipulating matter and energy to get meaning across to one another. We are doing this because we happen to be operating in the same language system, which we didn't invent, but if we didn't learn it, this would not be possible. And that learning process is not automatic. It has to come from mind to mind, even though the capacity to learn that is innate. And so what I'm saying is that just because I'm making standing waves of pressure in the air doesn't mean that those standing waves of pressure automatically mean something, okay? They don't. They were instantiated to have meaning when English was created. You understand what I'm saying?
Well, but even English is something that has evolved, right? So
yes, and that's great. But it didn't evolve outside of minds. So, and it didn't come from standing waves of air.
It's not the waves that are important, it's the concepts.
That's exactly my point. And the concepts are not generated from manipulations of air molecules. You see? Okay. They're not generated from that. But I can't get the concepts across without that.
Okay.
In this particular mode of communication, right? Mm-hmm. Okay, so that's, to boil down, I like your definitions of language because I don't, because we're boiling it down to connecting material and the immaterial and the, right?
I didn't say anything about anything immaterial.
A concept is immaterial. No?
Well, I mean, concepts exist in our brains. Our brains are not immaterial.
Okay, so then we'd have to get to philosophy of mind. But anyway,
and you thought this was going to be easy.
Well, it never is.
You were so excited.
Huh?
No, I'm just no. Go ahead.
No. I don't think it. I just don't have a way. Okay, so if we were to do that, like, so if we have to go there and say that, okay, a concept exists in your mind, but are you saying that your mind is your brain? So okay, so a concept for you is just a matter of pipetting out the contents of 5 neurons that are associated with that thought process. When we find them by functional MRI or, putting the electrodes there, we go, oh, when we tell him to think about this concept, these neurons fire, let's suck out the contents and put them in a pipette or put them in an Eppendorf. Oh my gosh, there is that concept right there. We have captured it. Do you agree that that is how that works?
No.
That's the matter that is that concept?
No. The concepts are an emergent property of the matter of the brain.
Okay, emergent property. So now this is, excuse my snarkiness, but this is more word salad to avoid actually getting nailed down to the idea that there is immaterial.
How clever of me.
Huh?
How clever of me.
You didn't come up with it. I'm not blaming you. I'm not blaming you at all.
Okay.
I just think that this does not help move the discussion forward, because if I were to
ask... Look, here's the thing. So you're talking about pulling neurons out of my brain. If you pull the neurons out of my brain, they're not going to work anymore.
So right? You can never understand that concept again. Is that what you're saying?
Yeah.
Wow. But that's not how we know the brain to work, actually. We don't know the brain to work in such a way that if you take out some part of it...
Well, you might be able to relearn it. But if you take... yeah. Because the concepts exist in other people's brains, too.
Because the concepts exist above our brains. It's transcendent over matter. Our brain is not producing the concept and transmitting it through electromagnetic radiation or whatever.
I can only get it from other people's brains. And their brains...
Okay, but what you're getting from other people's brains is it's not really coming from their brain, it's coming from their mind, and their brain is telling their mouth how to communicate the concept.
I don't see a difference between the brain and the mind.
Okay, well, I don't know so your philosophy of mind is that we are a bag of chemicals and there is no you. So you don't have a brain, you are your brain. This is what you believe. But this, I don't know, is tenable with your everyday life. Honestly, if you sat down and thought about it. it's not that you feel.
I have thought about it.
Okay. So when you think about how you come to making decisions,
Yeah.
you know, like do you struggle to make any decisions?
Oh yeah.
Do they just at the speed of depolarization just
Well, actually, sometimes it does.
Okay. But other times where it doesn't?
Well, normally when I have to engage my prefrontal cortex, there's a little bit of processing that has to happen there.
Why do you phrase it like that? When you have to engage in your prefrontal cortex, who's the you? Why don't you just say, well, my prefrontal cortex decides blah, blah, blah. We don't talk that way for a reason. There is a self.
Maybe you don't.
But you didn't either just now.
Yeah, so why do we talk about me? What do I mean by me?
Yeah.
Me is what exists behind my eyes. I don't know what it is.
Okay.
And I know it shuts off if my brain shuts off.
How do you know that? Has your brain ever shut off? Have you ever been dead?
That's every time that my brain shuts down operations, as far as I can tell, that's been what happens.
Okay, your definition of shutting down operations is a little different from what I'm thinking.
And everyone that I've ever known whose brain has shut down permanently, I've never been able to communicate with them either.
Okay, well, could another explanation for that be that they are just in a place where the mechanisms for communication that we enjoy here are not accessible? Like matter and energy, like we were just saying. We can come up with all kinds of explanations.
Mm-hmm. The sky's the limit. Come up with as many explanations as you like. But Occam's razor says it's most likely.
Okay, so this is good. I like the Occam's razor because I was thinking about this. When it comes to historical sciences or history, let's just say history. Like, I'm not a historian, but I'm wondering, is that really valid? Like, that when you have two possible explanations for how an artifact came into existence, that the simplest one is more often than not true? Because the way I understand history is that freaking anything can happen. People do crazy stuff, and we make stupid choices that are irrational. We have wars with each other, and we.
Okay, but here's the thing. So you're sort of moving in between possibility and probability, and those are two different things. Is it possible that some weird thing could happen in history? Absolutely. Lots of weird things have happened. I could win the lottery tomorrow. It's possible. Is it probable that I win the lottery tomorrow?
Okay, but we're not talking about tomorrow. We're talking about in the past.
Whatever. What is it probable that any particular weird thing has happened when we're looking at some event, right? So is it probable that, oh gosh, I don't know. Is it probable that an alien spaceship shot the first shot at Fort Sumter and started the Civil War? Yeah, that's possible. There's lots of weird things that have happened. It's not logically impossible that could be the case, but is that probable? No.
What?
So when we examine historical, what's happened historically, we have to stick as best we can with the probabilities.
Okay, but I didn't know we were talking about probabilities and possibilities with regards to Occam's razor. I thought we were just talking about simplicity. Isn't it the simplest answer is the best one?
Is the most likely.
Oh, okay. So is the most probable.
Yeah.
The simplest answer is the most probable.
So
I have to think about whether I agree with that, but sorry, go ahead.
Well, I mean, just take anything. Like, imagine that anybody comes to you and tells you any weird thing that has happened. Let's say that that I call you up and I say, "Oh my gosh, you're not going to believe it! I went to a basketball game the other day, and this guy on the one team, he jumped from under the other team's basket, and he stayed in the air all the way across the court and slam dunked on the other basket." It was incredible. Now, is it possible that that happened? Or is it probable that it happened?
Okay, so let me just answer what you're saying and then tell you that I don't know that we're talking about the same thing. I think it's possible, but it's improbable because in my repeated and uniform experience, people can't do that.
Right, right.
Okay, so
it's not possible.
But okay, but I'm not saying that. Like, I think I'm having a problem with the term simplicity. You know, like maybe what you do we always agree on what's simple versus complex? So what if there were a series of events that are all probable and there's like 26 of them that have to go together in order for event X to happen? And then there's this other way of explaining event X that only takes 2 events that had to happen. In that case, I'm thinking, and they're both, all the events are equally probable, nothing phantasmical, but the fact that you have to have each in this series, in one case, you need 26, in the other case, you only need 2. Would Occam's razor say the one where you only need 2 is more probable?
I don't know that I followed you at all there. Sorry.
Okay. Okay. What I'm trying to distinguish is between not the probability part, but the simple part, the simplest explanation. What is meant by that? Okay. And your examples were not given explanations that were simple versus complex. It was, implausible versus an outside of our everyday experience versus not. And that I don't think is the same plane of reasoning as simple versus complex. You understand what I'm saying?
Well, so what I was saying, and I'm glad we're getting back to evolution here. So with regard to evolution and common descent, what I was saying, the mechanism that I put forward is the mechanism by which organisms pass along their genetic information to their progeny. Right? That's very simple. We understand that happens. There's nothing really controversial about that. Agreed?
Yeah.
Okay. So if we understand that organisms pass along their genetic information to their progeny, then the similarity of sequences can be used to infer a ancestral relationship in the same way that I can look at my brothers, compare my DNA to my brothers or my cousins, or Charles Wesley. In fact, it turns out I'm actually related to Charles Wesley and John Wesley, the founders of the Methodist Church. They are my, I think, 7 greats uncles, believe it or not. And let's say I want to verify that. And I wanted to, in order to do that, I had to compare my DNA with the DNA of other people who claim ancestry from them. Then if we were more similar than anybody that we wouldn't expect to be in that group of people, then we can conclude more recent common ancestry than, let's say, me and Jonathan, for example.
This is a great example. This is a great example because it matches more with the problem that I'm trying to explain. Because you said that you would compare your DNA with the people who claim to have similar that ancestry with those people, right? With John Wesley. But what if you wanted to see whether they were telling the truth or not? How would you know that?
Well, you do the same thing, right? So if you, like, let's say that I met, this is how you disprove that hypothesis. So if my hypothesis is I am descended from John Charles Wesley's older brother, then if I were to compare my DNA with people who I know are claimed to be descended from John or Charles Wesley, they didn't have children together, and if my DNA was no more similar to theirs than yours or anybody else in an outgroup, then that would disprove that hypothesis. Do you see what I'm saying?
I don't really see how it would say anything about whether you were related, in fact, to Wesley.
Well, so again, it's not. You can't go back in time. You can't rewind the past and watch these things happen. You can't watch them have kids. but it's the evidence shows that it's more likely that that's the case than not.
It seems like all it really just says, without having to, I don't know. I feel like all it really says is that you are more related to those claimers of common ancestry to John Wesley than you are to other humans.
That's exactly right. That is exactly right.
But it doesn't say that you're both descendants of John Wesley. It says nothing about John Wesley. You know why? You don't have any genetic material of John Wesley.
But if we are more, if me and some other, let's say Jonathan is related to John Wesley. So if Jonathan and I are more closely related than anybody else, then yes, it does say that he and I have John Wesley as a more recent common ancestor than me and you.
But it doesn't say that at all. You have no information, genetic information about John Wesley.
If we're comparing our DNA with somebody who we know is descended from John Wesley, then yes, it does.
But how do we? Okay, but this is the point. We have an opportunity to know that somebody, you have a positive control, if you will, or you can anchor this set of equations so that you don't have too many unknowns, wherein you can say this person that has another avenue of information besides genes, they have heritage information, ancestry, like .com, right? They have historical data that says, oh, we go back and this was your dad and his dad and his dad. That's a whole separate type of information that we can say, okay, this person is now, let's take their genetic information and use it as a proxy for John Wesley's, because we don't have his, right?
No, we don't have his DNA, yes. But right. We have his descendants.
We have his descendants that we know are descendants because of history.
That's what I'm saying. Not
because of DNA, right?
No, that's what I'm saying.
Right, but what I'm saying is that we don't have that with us and chimps.
Okay, but that's a separate thing, right? So I'm not saying I'm not saying that humans and chimps have a common ancestor, and his name is John Chimpanzee. Right?
Why don't we? Who is the common ancestor? That was a person, if they existed.
Okay. So this is where maybe I wasn't clear. So I'm not saying that we can point to an individual organism and say that is the most recent common ancestor when we don't know who that is. So in the case of the analogy that I was making with John Wesley, we know who that is, and we can compare my DNA with somebody else who we know is related to them, and we would see what we would see. If we don't know, so let's say that we don't know anybody. So let's say that you and me and Jonathan, we show up in a room together and all that's there is a DNA sequencer and a bunch of cotton swabs. And we say, well, we got to pass the time somehow. So let's sequence our DNA and find out who is more closely related to whom. So I sequence my DNA, Jonathan sequences his, and you sequence yours, and we line them all up. And it turns out, to everyone's surprise, that you and I are more closely related than either one of us is to Jonathan.
Mm-hmm.
Now, it doesn't tell us anything about who that ancestor is that we have in common. Clearly, there is somebody, right? There has to be somebody that we have in common. I don't know who. Doesn't really matter who. But there is somebody that we have in common that is an ancestor to both of us that is not an ancestor of Jonathan's. And that is what I'm saying about humans, chimpanzees, gorillas, I forgot, orangutans again. That's what I'm saying. Does that make sense?
No, I mean, because again, you are like smuggling in this concept that this process is applicable across to other species. And I'm saying,
why wouldn't it be? Why wouldn't it be?
Because you don't get the rooting in history that we have. When I say history, like I was saying before, that you can say we have evidence from our repeated experience that humans make more humans. We do not have any evidence that humans make something other than humans.
We're not looking at that. We're not looking at what humans make. We're looking at DNA. All we need to know is that organisms, it doesn't matter what we call them. All we need to know is that organisms pass along their DNA sequences to their progeny. That's all we need to know. That's
okay. That is all well and good. Are you aware of orphan genes?
Tell me about orphan genes.
Okay. Well, orphan genes are essentially, they're written like capital ORF and then AM as a play on open reading frame, and also the concept that they have no parentage in the way that we would expect given Darwinian concepts of all genes come from older genes because of common ancestry, descent with modification, those ideas. But as we are doing more genome sequencing projects, this was found earlier as well, but there were a lot of these genes that have no homology to any other sequence in any other organism. And so they were actually thrown out at first as mistakes, and whatever you want to call them, like sequencing mistakes. Even though the sequencing was accurate, it's just they assumed that they weren't actual gene products, even though they were considered open reading frames. They had the signatures of such. And so lo and behold, as sequencing gets cheaper and we do more and more genome sequences, we find that these things pop up more and more in each genome, and they are also called taxonomically restricted genes. So where they show up in some narrow slice of a branch of the tree of life, and they don't show up anywhere before or after, or I shouldn't say after, but anywhere like more basal. And the question is, where do these come from? Like, how can we support the existence of these within a framework that says that genetic material is just delivered down to the generations with modifications, not with total novelty? You don't take gene A and say, it's not, the polymerase doesn't get creative and just change the gene.
No, the genome is the Wild West. Are you kidding me? We just got finished talking about all these weird things that can happen with chromosomes.
Right, but that's not the same thing as coming up with de novo sequence that has no relationship, no detectable relationship to any other sequence. This is not the same thing as taking chunks of DNA and mechanically moving them to other positions in the chromosome.
Well, if that's true, then I don't know where it comes from.
Okay, do you agree that it's problematic for common ancestry?
So are there a lot of these that are in the human genome that we don't see in the chimpanzee genome?
Oh, I was going to look that up. Jonathan, do you know the answer to that? I feel like...
The answer is actually yes, I believe. There was a recent paper in Nature, actually, and this isn't to do with the human genome in particular, but this one comes to mind. There's a recent paper in Nature published by some biologists in London, and according to this paper, they sequenced the ash tree genome. And within it, they found 38,852 protein coding genes. And of these, 1/4, which is 9,604, were unique to ash. And so that's 1/4 of the genes which share no homologs with any of its close relatives. Now, there are papers which also look at the human genome, but I don't have them off the top of my head. I need to go and dig them out.
Yeah, all right, I'm going to share the link. Oh, I'm sorry. How do I get there to that? I was going to share this link that I just got from an article written by Ann Gager about human orphan genes. And I think it will, yeah, so that can be a resource. But yeah, it looks to be that there are. And another interesting thing about this problem is that you would think that the more genomes you sequence, the more you'll be able to account for what was previously considered an orphan gene, that you'd be finding their relatives in more organisms that are getting their DNA sequenced, so that the total number would be going down. This is what you would expect, that the problem is not that they're genes that can't be explained by common ancestry. It's just that we haven't sequenced enough organisms to find out that they have plenty of homology to be found, right? but the total number of orphan genes actually keeps growing sort of linearly. It's not tapering off. It just continues to grow with each genome that's sequenced, because they all have their own orphans.
Well, if this is a real biological phenomenon, then that's exactly what we would expect.
What's the phenomenon that you're why is that what we would expect? I'm
saying if this is a biological phenomenon as you're describing it, that genomes develop their own de novo genes by whatever mechanism, I don't know what. But if it's something that we would expect to see in every genome, then as we're constantly sequencing genomes, so we would expect to see more of those. That's not surprising.
Okay, I think that's good logic. And
I think actually you would expect to see less of them, because the more genomes we sequence, the more homologs we should be finding for those apparently.
But not if this is a real de novo mechanism. If it's a real, as Charisse is describing, if it's a real de novo gene production sequence, then we would expect to see more of them the more genomes we sequence.
Okay.
Right. I'm submitting to you that back to the question of whether the modes or whether the activity of a conscious agent in the past is detectable, that this phenomenon is an example of such a thing, that what we are detecting is the activity of a conscious agent in the past instantiating genetic biological information, specified information from scratch.
That's possible, but I don't know that it's probable.
Oh, okay.
I mean, look, intelligent. I'm not saying that intelligent design is not the case. It very well may be the case.
I think it's the best case.
Well, nothing that I've presented is inconsistent with intelligent design.
Yeah, well, okay, I have to think about that, because
it's just that if there was a designer, the way things were designed look a lot like they had common ancestry. You could design that, right? So just like you're presenting me the hypothetical of an alien creating me and implanting me in my mother, for example. So I would look like I was born from my parents when I'm actually not. So you could design that. You could design a situation whereby something looks like it kind of developed naturally, but did not actually happen that way. That's possible. It's absolutely possible. I'm not contradicting that.
But these phenomena don't look like they happen naturally. Orphan genes do not look like any natural process. We don't.
Not that we know of. No. I mean, I don't know. I mean, I'd have to look into it, I guess. Maybe see what's out there.
But they look like a conscious process.
I'm not sure what that looks like.
I'll tell you. You know what I did my PhD on? DNA repair. You know what I had? A DNA synthesizer in the lab. And there was a computer, and I could type in exactly what sequence of DNA I wanted this thing to chemically produce. Out of my mind, I told matter what to do. I controlled the conditions, and I made specified information embedded into matter, and I used it, made crystal structures, mind over matter. That is what I'm talking about.
Well, here's the acid test then. What I would suggest is that you why don't you do this? If you take some model organism, let's say, and start putting genes into it, if you could synthesize new genes into it, put it out there into the wild, let it be discovered, have some other team characterize it. If they are able to take your if they're able to detect your inserted sequences as these orphan genes, then you've kind of proved your point, I think.
That's an interesting proposal. I have to think about that. Although I'm not saying that there's anything quintessential, there's anything essentially different about the information that orphan genes are versus homologous genes. That's kind of precisely my point, is that I'm putting them in the same category as biological information. And I'm claiming that the homology that we see is being misinterpreted as common, as being sourced from common descent, when it's really common design. And then there's these cases where there's no feasible way to assign common descent because of the orphanness, and that makes it a stronger argument.
So here's what you do. You design an organism and you put it out there and you let people try to place it in evolutionary history. And then once they do that, then you write your paper up and get it published in Nature and say, see, I fooled you all.
That would be really brilliant. Do you think that they would accept that? Do you think that that would debunk the whole thing?
I think they'd be pissed off about it, but yeah.
Actually, Zachary, there's a paper which comes to mind. I just dug it up. This one is a study of bacteriophage, X174, and I'm going to read the abstract and ask you to comment. Replicate lineages of the bacteriophage X174 adapted to growth at high temperature, and either of two hosts exhibited high rates of identical independent substitutions. Typically, a dozen or more substitutions accumulated in the 5.4 kilobase genome during propagation. Across the entire data set of 9 lineages, 119 independent substitutions occurred at 6 to 8 nucleotide sites. Over half of these substitutions, accounting for 1/3 of the sites, were identical with substitutions in other lineages. Some convergent substitutions are specific to the host used for phage propagation, but others occurred across both hosts. Continued adaptation of an evolved phage at high temperature, but on the other host, led to additional changes that included reversions of previous substitutions. Phylogenetic reconstruction using the complete genome sequence not only failed to recover the correct evolutionary history because of these convergent changes, but the true history was rejected as being a significantly inferior fit to the data. Replicate lineages subjected to similar environmental challenges showed similar rates of substitution and similar rates of fitness improvement across corresponding times of adaptation. Substitution rates and fitness improvements are higher during the initial period of adaptation than during a later period, except when the host was changed. And so here we see an incorrect evolutionary history generated by these phylogenetic algorithms because of these independent convergent substitutions.
Yeah. But we're talking about bacteriophages, right? So these are very tiny genomes. Mm-hmm. I could see something like that happening in something like a bacteriophage. Where this modification or the convergence was happening in laboratory conditions, or yeah. So I can see that you could. A bacteriophage is such a malleable life form, I guess. I can see that being subject to really strong evolutionary pressure in the lab, such that you sort of completely distort. It's kind of the opposite of what I was suggesting to Charisse. You're sort of taking two things that have descended in separate ways under maybe similar pressures, and now you can't. It's the evolutionary history has been obscured. Mm-hmm. Absolutely.
But the point is that the evolutionary algorithms are designed to produce a tree. So irrespective of the degree to which there are deviations from the supposed nested hierarchy, your algorithms are always going to produce you a tree.
Sure. Yeah, absolutely.
And so what's your null hypothesis?
In that situation?
Well, what's your null hypothesis? Because my null hypothesis is uncommon descent. Right, so you have to allow that as a possibility in order to
what does uncommon descent mean?
So if you take a sort of intelligent design view where different taxonomic groups are created de novo, then that is uncommon descent, where you do not have we do not all trace our origins back to a single progenitor.
So taxonomic groups are created de novo.
Right.
Which taxonomic groups?
Yeah, don't use their term.
What do you mean exactly by that?
Why do you want me to use taxonomic group trees?
Well, because the taxonomy is actually a process of comparing similarities and differences and grouping them using mathematical graph theory or whatever.
Sure, but when I talk about taxonomic groups, I mean things like phyla and orders and classes and genera and species.
But
with their arbitrary categories.
Right. Well, she's right, but I'm asking, like, when you say that they're created de novo, do you mean like the entire animal kingdom was created de novo?
Right. So for instance, if we took the view that that humans are created independently of other primates, for example, or that you know, and so on. So I mean, is that your null hypothesis? Because it seems that, I mean, what would it take for you, based upon the molecular data, what would it take for you to say, okay, common ancestry has been falsified?
Well, so here's an example. So we saw the way that the sequences were laid out, like for example for cytochrome C, right? Mm-hmm. If every single species had a completely different sequence for cytochrome C, mm-hmm. which they could, you know, they absolutely could have a different species or a different sequence for cytochrome C, then that would absolutely disprove my argument.
So there's a paper published in Trends in Ecology and Evolution, which looked at the cytochrome B gene. Are you familiar with this?
No.
This is this quotation directly from the paper. It says the mitochondrial cytochrome B gene implied an absurd phylogeny of mammals, regardless of the method of tree construction. Cats and whales fell within primates, grouping with simians, monkeys, and apes, and steppe hinds, which are lemurs, bush babies, and lorises, to the exclusion of tassiers. Cytochrome B is probably the most commonly sequenced gene in vertebrates, making the surprising result even more disconcerting. So you think, it seems to me that you're cherry picking. I mean, if you look at one gene, it gives you one phylogeny. You look at a different gene, it gives you a different phylogeny.
No, that's not the case. In fact, I've looked at, usually what I look at is, I've done this talk before with a different gene, apolipoprotein E, which is the one I studied when I was getting my PhD. And apolipoprotein E has the exact same pattern as cytochrome C. And I would guess that the more genes have that pattern than not. I would guess that cytochrome B is maybe an exception.
So at what point do deviations from the hierarchy become evidence for uncommon descent?
That's a good question. I don't know.
Because it seems that you would have to establish that in order to make a positive argument for common ancestry. Otherwise, it's unfalsifiable and it's basically a tautology.
Well, no, not necessarily. I mean, if you look at the entire, you can do the same. You can create the same trees using genomes as well, right? And how does that turn out?
You can do the same using genomes, yeah. And there are different. There's a lot of incongruity in the phylogenetic record. In fact, that's the reason why there's so many papers on attributing these deviations from the hierarchy to convergent evolution, to lateral gene transfer, even among higher life forms. To incomplete lineage sorting, and so forth. And there's these mechanisms that are invoked as ways of getting around the fact that there are so many deviations from the hierarchy.
But they're deviations from the hierarchy. They're not the main situation, right? So it's much more common. It's much more common to find genes arranged or the sequences arranged in a hierarchy. Yes.
I would suspect to be the case that if one was to look at, I suspect that there is an evolutionary plateau at one point where there is no hierarchy. So, so for instance, you talked about, for instance, so the molecular variance patterns. If one can show there's a necessary hierarchy for, say, primates, but if you extend it to all life which possesses that particular gene that we're talking about, I suspect that beyond a certain point, there's probably an evolutionary there's probably a plateau where you don't find that necessary hierarchy.
Well, I mean, so we kind of see that with cytochrome C, right? So once we get down to yeast, the sequence is almost completely divergent from the human sequence. And that's fine. And listen, that's not the point though. The point that the thing that gets under everybody's skin is humans and chimpanzees.
Okay, but what would you say is the strongest line of evidence for universal common ancestry? Not just humans, chimps, and other primates, but universal.
Everything I've laid out. For the protein sequence, the nucleotide sequence, the transposon evidence, everything.
So at what point does a deviation become evidence against? I mean, how could you disprove that from the molecular data?
Well, again, as I've said, if every sequence that you compared was random, was different, If there was no hierarchy possible, then that would be a total disproval.
So, I mean, we talked about orphan genes earlier, and
but that's not every sequence.
Yeah, I'm aware of that, but there is a substantial number of them, and they are growing. But the problem is even more profound than that, I think, because I mean, for example, I study the cell cycle, mitotic cell division.
Sure.
And when I look at the proteins that are involved in mitotic cell division, there are virtually, for almost all of them, there are no homologs among prokaryotes.
Okay.
You have a quantum jump.
But we're talking about prokaryotes versus eukaryotes. I mean, that's
right.
a huge difference.
Yeah, exactly. But you cannot trace. I mean, there are moments in the history of life when you see tremendous innovations where you have proteins that emerge de novo and have no evolutionary parents.
Maybe. Look, yeah, it's possible. So maybe there was some designer involved between prokaryotes and eukaryotes.
Right.
I don't know.
That's
maybe.
Concession.
But the key difference, right? So this, and I'll give that up. Listen, I will happily give that up if everyone gives up the collapse in the difference between chimpanzees and humans. How about that?
If what? You'll give up what?
I'll say that there's the development the genesis of the eukaryotic group was designed if everyone else agrees to give up the special creation of humanity.
I mean, science is not a negotiation.
I'm trying. I'm putting everything out there.
Is this what we have come to?
Maybe. I'm just trying to get along.
We are getting along.
I'm outnumbered here. Come on, give me a break.
I'm sorry. You're right. I was cognizant of that, so I was trying to,
Yeah. Zachary, I mean, one of the issues as I see it in evolutionary biology is that evolution is a theory not so much of similarity but of transformation. And so, yes, it's nice to be able to put all these proteins and genes and so on into a nice hierarchical distribution and try to infer phylogenies, or you get all these eyeballs out as Darwin did. You slap them down on a table and draw arrows between them and say, hey, one evolved into the other. But can you actually propose a mechanism by which you can accomplish the A to B transitions? Because as I see it, I mean, for instance, the fossil record is very good at documenting, the amount of time that is that one has in order to scale these A to B transitions. And we look at irreducibly complex macromolecular machines in the cell and so on. These seem to suggest that the neo-Darwinian mechanism is insufficient to produce biological complexity. And so the issue of scaling the A to B transitions and explaining the differences between organisms seems to be, to me, far more interesting than simply explaining the similarities between organisms.
Well, that's a different question altogether, right? So the question of how did it happen is different from the question of did it happen? And so what I've presented here today is evidence in support of the fact that it happened, right? So if what we look at in the sequences is true, then the explanation of it happened because of common ancestry is more likely than the alternative. So that's not the how. And so I just want to make clear that I've presented the how, not the why. And so the why is don't know, right? So all we can do is infer, as Sharice was saying, all we can do is infer from the uniformity of what we see that we have organisms, accumulating mutations, other differences, weird things happen with the chromosomes sometimes, and passing along those things to their progeny. We can see organisms being acted on by their environment. We can see organisms being separated by things in their environment, and we just all we can do is infer from there, right? Just like Darwin with the glyptodon and the.
But is it an adequate tool to? Is the extrapolation from that warranted? I mean, in order to get from a single cell's organism into the diversity and complexity of life we see today, we need adequate resources, right? And yes, we see these processes occurring, and this is one of the take-home messages I took from the Royal Society meeting, which occurred recently in December in London on evolution, was they proposed all these mechanisms and documented that yes, these mechanisms occur and need to be incorporated into the modern evolutionary synthesis. But what was lacking was a demonstration, either empirically or theoretically, that those same mechanisms actually were adequate to explain biological complexity. For instance, how do you assemble a developmental pathway from a zygote or fertilized egg to a viable form of the organism? Because you need to traverse a distance, and it's no good to stop halfway along that distance because your organism's dead. You have to traverse all the way from A to B. And how do you put that together by means of an evolutionary stepwise pathway? It seems to me to strain credulity. Same with macromolecular machines. How do you put together molecular machines which require multiple functions to be performed in order to accomplish an overall purpose in the cell?
Again, I don't know. I'm not presenting the how. I'm just presenting the what.
Because it seems to me that if we cannot propose a mechanism by which evolution could occur, how can we be so sure that it did occur? And maybe
because the evidence demonstrates that it did occur, right? So the question of how it occurred is separate.
Right, exactly. But it raises questions as to whether perhaps you're misinterpreting the evidence.
Maybe.
Because when it comes down to, there's explanatory scope and there's explanatory power in this plausibility, and these are the means by which, the tool set by which we judge whether an explanation is the best. And if there's no viable naturalistic mechanism that can produce it, then I'm not so sure that we can be so confident in these arguments. And maybe there are other explanations for that data. So that would just be what I would say. I think there's, and in any case, even if your arguments are completely valid and sound and demonstrate common ancestry, once you move away from a gradualistic neo-Darwinian evolutionary account of life's origins and development on Earth to a more sufficiency view, where you have sudden jumps and bounds in evolution, then which are aided by an external agent, then the boundary line, I think, between gradualism between common ancestry and special creation starts to become somewhat blurred.
Maybe, and again, I've not said that special creation is impossible, given what I've done, what I've shown, or at least it's not inconsistent with it necessarily.
But out of interest, do you have any reason to think that the neo-Darwinian mechanism is sufficient to explain?
Yeah, I don't have any. Is the neo-Darwinian explanation insufficient?
Yeah, that would be my claim. And
yes, it absolutely is. Almost every scientific theory, every scientific discipline is insufficient. Yes, absolutely. We need to keep developing it.
But it depends how broadly you define the term neo-Darwinian. But let me try this again. Do you have reason to think that materialistic processes are adequate to explain biological complexity?
Do I have reason to think that? Yes.
What would be that reason?
Because all I know are materialistic processes.
But so you haven't had any convincing demonstration, empirical or theoretical?
Well, here's the thing. So I've already demonstrated that common ancestry is the case. All I know of in terms of processes that can take place are materialistic. At least the only ones that I can demonstrate, the only ones that I can describe, the only ones that I can wrap my head around are materialistic. Okay. So
that's super honest.
Until I get to the point where I can have something else that I can describe properly, it's just that's the only thing that I can rely on at this time.
What do you think, Zachary, of the problem of reductive evolution? The problem that often, one of the main mechanisms by which evolution is thought to proceed is by duplication and recruitment of genes, right? so as a gene becomes duplicated, and then one copy is free to explore sequence space, as it were, in search of some new function. But there's the problem of reductive evolution that because of especially in bacteria, where there's a pressure for small genome sizes, it's more evolutionarily favorable to discard and get rid of the duplicate copy, to purge it from the genome because of the energy cost of transcription. And there have been studies which have shown this to be the case. For example, there was a paper by Ralph Silke and Gager and others on looking at the TRPA gene, where basically they incurred two mutations in this TRPA is one of the genes involved in the biosynthetic pathway of tryptophan, the laurus amino acid. And one of those mutations that they incurred partially inactivated the TRPA gene. The second one that they incurred fully inactivated it. And that seems to be a very easy pathway for evolution to take, where it just needs to take a two-step pathway. One partially activates the gene, the second one fully activates it and gives it full functionality back. But in none of the lineages of E. coli in their experiment was that path taken. In fact, what happened was that they purged the broken gene from the genome because of the energetic cost of transcription. And that's not the only study which has borne that out. So that's a significant hurdle for the evolutionary mechanism. And also, Doug Ack's work on protein structures and what it takes to produce new protein structures, the rarity and isolation of stable and functional protein structures in combinatorial sequence space. I mean, what would you say to those sorts of objections?
So you're describing E. coli that's trying to maintain a small genome, essentially. Yeah. Mm-hmm. And so you're saying that contradicts evolution because it's not becoming more complex?
There's a time limitation, a very severe time limitation because one of the main evolutionary mechanisms that's put forward is that new genes arise by duplicating old genes and then recruiting of the duplicate copy, right?
Well, that's one thing that can happen, yes.
Yeah, and it turns out that I'm talking about bacteria specifically because bacteria have there's a selective pressure for small genome sizes. That there's because of the energetic cost of transcribing a broken gene, these are typically purged from the genome rather than exploring sequence space, even when there's very modest changes that need to be made to incur new function.
Well, but you just described, of course, they have to keep their genome small.
Yeah.
So that's tremendous pressure in the opposite direction. I mean, the evolution of prokaryotes, versus the evolution of multicellular organisms, of course, is very different.
Mm-hmm. Yeah, that is one point. But there's a whole range of points. I mean, what would you say about the problems of population genetics? For instance, I mean, you're probably familiar with the waiting time problem.
No, describe it for me.
Okay, so waiting time problem. So population genetics, as you know, I'm sure, is the mathematical expression of neo-Darwinism. So basically, the equations of population genetics are used to infer how long it would take you to acquire a mutation given the population sizes, the generation turnover times, the average mutation rates, right?
Okay.
And one can perform that analyses and look at the fossil record as a means of documenting the amount of time, geologically speaking, that you have to get from the A to B. So, for instance, one example in the fossil record is the evolution of the whale, which evolved from terrestrial mammals, and you go from Ubiquitous into Hyas, Angulocetus, Protocetus, all the way down to Dorodon, Basilosaurus, within a very narrow window of geological time, in the space of a few million years, 3 or 4 million years. And then we look at the equations of population genetics, and there was a paper published in 2008 in the Journal of Genetics by 2 mathematicians called Rick Jewett and Dina Schmidt, and they were attempting to refute Michael Behe's work in The Edge of Evolution, where Behe, based upon empirical malarial data, had shown that to accomplish a 2-step coordinated change, you would require about 10 to 15 years for that to occur. But Jurt and Schmidt, according to their theoretical model, suggested that it would only in fact take in humans, assuming a population size, an effective population size of 20,000 individuals per generation, that it would take to accomplish a two-step coordinated change 216 million years, which obviously is prohibitive given the 6 million year transition time. But in whales, the whale transition, Rick Sternberg, who's an evolutionary biologist, took the equations from that Jurt and Schmidt paper and redid the math to say, okay, we're dealing with more individuals and so on. And according to his calculation, it would require about 43.4 million years for that A to B transition to occur. But according to the fossil record, you have much, much less than that. And you have a lot of changes that need to occur, especially in one example that he highlighted in particular was the evolution of an intra-abdominal current heat exchange system, because the testes in whales are actually inside the body, right next to the muscles generating heat during swimming. So you have to actively cool down the gland, and so you need an intra-abdominal current heat exchange system. So if you imagine that just 2 mutations are necessary to accomplish that transition, coordinated changes that is, then you require a lot more time than is afforded by the geological record.
Okay, so what's the alternative then? That somebody came in and consciously tweaked the whale evolution?
Well, it's not simply that is a so lone example where it's the anomaly and actually everything else is all hunky-dory for Darwinism, but rather that seems to be the norm rather than the exception, that there's just not enough time for, mathematically speaking, for neo-Darwinism to work.
I don't know. I'm not a population geneticist. I'm not sure.
Are you getting exhausted? Because this should not be like, an interrogation, on some deserted island.
Actually, I will have to take my leave in just a couple minutes.
Okay.
If you don't mind.
One quick question before you go, if I may. What's your view on the topic of irreducible complexity?
And say that.
What is your view on the topic of irreducible complexity?
Irreducible complexity.
Yeah.
Yeah. So again, this is a concept, Michael Behe's concept, correct? Mm-hmm. Yeah. So the thing about that is, this is another one of these concepts. I think kind of like the concept of species that is kind of human derived. It's sort of by the way that we look at the world. I'm not sure that it's scientifically validated.
Okay, so I mean, just as a very quick example, one thing that, because I love cell division, I study cell division as a PhD,
Mm-hmm.
but in bacterial cell division, which is equally fascinating, there's the bacteria divide by binary fission, which is of course a very different process from mitosis, which we ourselves divide by. And in binary fission, the cell in a rod-shaped bacteria will elongate to about double its original length, and then you have a bifurcation in the middle, the formation of the division septum, and it'll bifurcate in the middle into 2 daughter cells. Now, you have to elongate in the process the bacterial cell wall, which is made of peptidoglycan. And this is done by enzymes known as autolysins, which have to be coordinated in a very sophisticated manner in order to prevent cell death. But at the same time as you're severing peptidoglycan cell wall, you also have to reconstruct the bacterial cell wall. Otherwise, due to the osmotic pressure, the cell's going to burst and lyse. So you need a mechanism for rebuilding it, and you have a protein called bactoprenol, which is shuttling the peptidoglycan precursors across the cell membrane to the cell wall, and you have the penicillin binding proteins, which are involved in the transpeptidation process, the peptidoglycan cross-linking. Now, those penicillin binding proteins are in fact a target for penicillin antibiotics, as their name suggests, because the bacterial cell wall reconstruction is a target for these antibiotics, because we understand that if you play with that, then the cells dies because it's so built. And so how do you put together, in evolutionary terms, a mechanism to sever, rebuild in a very coordinated manner? It seems to defy evolutionary logic.
Not necessarily. Again, we don't necessarily know how all these things came about. But if we look at the evidence, that we can see that common descent is the most likely explanation. I mean, I'll just repeat that again. So I don't know exactly. I mean, you can take any sort of mechanism, any sort of biological system in isolation is quote unquote irreducibly complex, right? So how did we evolve lungs? If we didn't have lungs, then we couldn't breathe. So how can how can you evolve a human if you don't evolve the lungs first, right? So that must mean that it's irreducibly complex, but
it is.
It's not necessarily the case, right? But you can take anything, like our hands, or our feet, or our heart, or whatever. But we don't evolve these. At least it doesn't seem to be that these systems come about separately. That they're all kind of a package, and the entire package is evolving together, and the entire package is under evolutionary pressure at the same time. And there may be scaffolding systems that are in place, that have been in place in the past, that are not being used right now. For example, we have an appendix that exists now that really doesn't have much of a function in humans, but did in the past. So we know that those vestigial things can happen. There can be vestigial remnants in our...
Sure, but it's difficult to conceive of any sort of scaffolding type process that could account for that, or even bacterial flagellar assembly. I think is very difficult to explain in evolutionary terms. The process of embryonic development, where you're going from an A to B transition, namely from the zygote to the viable form, because Paul Nelson coined the term ontogenetic depth, which is the distance that you have to traverse from the fertilized egg or the zygote to the viable form. Whether it's C. elegans or Drosophila or any other organism, if you go 3/4 of the way across, you're a dead organism. You have to go all the way across. But how do you put that together in a stepwise neo-Darwinian fashion? It seems to me to be very difficult to envision.
Yeah, it might be difficult.
Or metamorphosis, which even compounds the problem, where you go from caterpillar to, within the chrysalis, a kind of soup-like thing, to being reconstituted into a butterfly. So you need to have two body plans in view, and so you need to have some goal in mind. And a goal-directed process seems to me to be what you require in order to go, in order to accomplish irreducible complex systems, or process, or systems where you have an end goal in mind, like embryonic development and like metamorphosis. And a goal-directed.
No, you don't need a goal. You just need a strong selective pressure.
No, you need a goal, because you cannot select in a stepwise manner, because you need all of the things.
Right, but you're assuming that there's a teleological process there, and there might not be.
But when you're evolving a developmental pathway, you can't select it gradually, because you need the entire process to be there.
But you're assuming that the process is the end goal, and it's not necessarily the goal.
So, I mean, why would natural selection, for example, select for a dead caterpillar? It has to have an end goal in mind in order to direct it to become a butterfly.
Why would it select for a dead caterpillar?
Because to go from a caterpillar to a butterfly, which is the process of metamorphosis, which we know happens, sure, you have to dissolve the tissues and organs within the chrysalis, and then it gets reconstituted and it becomes a butterfly.
But it's not a dead caterpillar.
No, but why would natural selection select for the caterpillar in the chrysalis stage, where it's not going to live in that state? It needs to.
You're asking why? I don't know why.
Right, but it seems to be
the wrong question.
Because if you're going to demonstrate that evolution is a viable theory, you also need to have some explanation for how it's even conceivable or viable. And it seems to me quite plain that it's not, from all kinds of evidence. And we haven't even talked about the information beyond DNA, because you talk about the molecular data, well, much of the information necessary for driving the morphogenesis of organismal form actually exists beyond the purview of DNA. For instance, in the ion channels, which establish electrochemical potentials during development, although these ion channels are themselves encoded by the DNA, their spatial distribution is not. There's the sugar code, which is involved in development as well, which is beyond DNA. So it seems to me that if you want to explain the evolution of novel body plans and evolution, mutating DNA is simply the wrong tool for the job, because you're not, no matter how much you mutate your DNA, you're never going to be able to produce new body plans.
Well, it might be an insufficient tool for the job. There might be other mechanisms at play, for sure. I'll grant you that. I don't know what
mind.
Maybe.
This has been so wonderful. Look, we all are a big happy family, and we agree. There's evolution, there's mind. They're both happening.
I will agree with that. There are evolution and there are minds.
Yes.
Okay, we don't agree.
All right. Well, hey, Jonathan, it's been a pleasure, and Charisse, it's been a pleasure as well.
Likewise. Thank you for engaging. And
thanks for the invitation, Jonathan.
Yeah. Thanks, Zachary.
All right. Cheers.
All right. Take care.
Bye.
Bye.